Yeah, no, she's super nice, super smart. Like actually, I made some of the so. All right. Well, yeah. Well, okay. Yeah. But another random connection. Really? Yeah. Yeah. What is he doing? I don't know. That's so fun. You gotta cut me in. It sounds really fun. Yeah. You know he's doing that, right? I just not know that. Oh, okay, great. Yeah. So he doesn't Oh, I love to be. Yeah, all you. Yeah. Cool. All right. Well, I won't distract you. Yeah.
Hi, everyone. We're going to start again. I'm Margot. I'm a fourth year PhD student in the Lynch Lab. I study the oral microbiome, and I'll be your moderator for the rest of the day. Our first speaker today, this afternoon, is Dr. Gabby Fredridakis. She is an assistant professor of medicine in the school at UCSF. She received her PhD from Stanford. Gabby heads the data science co-lab at UCSF, and her research uses computational approaches to study patient immune state using SciTalk and single cell sequencing in a variety of contexts, including infection, cancer, and steady state. So please welcome Gabby.
Wonderful. Thanks, Margot, for the introduction, and thank you all for coming to listen, for the invitation. I'm really thrilled to get to speak to you today about some of the work that we've been doing in the space of integrating microbiome and immune system data. So what I'd like to do with the time I have ahead is sort of to start out with the motivation for really trying to take all this high-dimensional data from the microbiome and the immune system and see what kind of inferences we can make from these complex data sets. So discuss sort of very briefly some of the types of approaches to this kind of integrative analysis, and then tell you a little bit about my work as a postdoc, putting together microbiome and immune data in response to dietary intervention, though followed with a recent project that's been more around sort of directly trying to do this integrative analysis to look for shared variation across the immune system and the microbiome. Okay, so first, I know some of the attendees today, maybe this will be preaching to the choir, and others work in very different types of systems, but hopefully I can convince you as well. There's sort of utility to doing this kind of work of high-dimensional profiling of both the microbiome and the immune system. So why are we doing this, and why are we doing this now? These two systems very much shape one another. What we've seen both from mouse models and association studies in humans is very much that there is significant crosstalk, and that there's really, in sort of early development, these systems are shaping one another, are in constant conversation. And importantly, that what's really been emerging is how the microbiome is very much a mediator of many sort of immune foundational diseases and sort of chronic diseases that are really coming to bear in sort of industrialized society. And so this becomes incredibly pertinent, as well as an incredible opportunity due to the malleability of the microbiome and sort of how we can continue to use those connections ultimately to guide human health. And this is a particularly exciting time to be working in this area because all the folks in sort of technological development around data generation have really given us a whole menu of choices for which to really get complex pictures of both the microbiome and the immune system. So everything from sort of next generation sequencing to be able to look at sort of who's there on the microbiome side, what genes are there, what they're expressing, and then ultimately sort of what they're producing around metabolic output. And then conversely, sort of on the immune side, we can get, sort of analogous methods in certain types of ways, though sort of with very different approaches to look at cell type composition, what type of host cells are there, the types of activation levels that we're measuring, and then sort of production, everything from metabolic signals, but then also cytokines and other types of communication. So all this is, what gets me out of bed in the morning. Is very exciting, but it does come with this big challenge if you are lucky enough to get all of this data from a given system of how do we start to integrate this complex data such that we can actually learn any sort of biological phenomena from this large mass of data. So I like to think a lot about data integration strategies, and that's really sort of a central tenet of the work that I do, both in sort of the immune and microbiome context, but also sort of more broadly in just looking at states of the immune system and how we put data together. In the context here of the microbiome and the immune system, I sort of like to think of 3 different buckets of ways that we can do data integration, though this is not exhaustive. So the first is, I think, the first thing that we think of, and maybe the first thing that's tractable, is just sort of taking pairwise relationships of host immune features. So if we extract a bunch of immune features, extract a bunch of microbiome features, let's just like correlate them pairwise, look across and just see like what relationships emerge, or you can use some fancy version of correlation to do something similar. And you can get a lot of information that way. But you are sort of selling yourself short if you look sort of just there. And so fortunately, there are other approaches that we can also take to be able to sort of take advantage of the multivariate nature of our data. So sort of a next class of methods are multivariate models, right? So we can take what's been developed in other fields around taking into account a large number of features and potentially the interactions between those features, or sort of the cumulative evidence from those features, to be able to predict an outcome of interest. So you could either take all of your microbiome and immune features, put those in a multivariate model such that you have then and some outcome variable of interest potentially, and look for a signal there, or you could collapse down on one, and if you have a particular immune endpoint that you're interested in, feed in your microbiome type features of a multivariate model. And the one that I'll focus on actually most is this idea of sort of identifying shared components across the microbiome and the host immune system. And these are really sort of in a class of dimensionality reduction strategies of really trying to find axes of shared variation that are sort of picking up a sort of more tractable set of sort of meta features or factors that we can then both use to make inferences and then also sort of peel inside and say, okay, what of our underlying immune and microbiome features are contributing to this sort of shared variation that we are capturing across our data set. And that will be through the latter half of the talk. So first I wanted to go over some of the work that I did as a postdoc was dipping my toe into this idea around what can we learn simultaneously about the microbiome and the immune system. And this was work done in the Sonnenberg lab and really sort of built around this central hypothesis that over time and with industrialization, there have been big changes in the way that we live, the foods that we eat, what we're exposed to, and that while this comes with many benefits, this has resulted in a series of changes to our microbiota that we can infer from different types of studies. And sort of in parallel, we see this increase in sort of immune-mediated disease. And this open question of, can we bring about certain lifestyle changes? The one that we focused on here was diet, such that we can reshape the microbiome diversity and metabolic output to then confer some degree of protection or mitigation from sort of inflammation-based chronic disease. And so to test this idea so it's a very grand idea, but to sort of look at it in a somewhat controlled way, though still in a complex human system, we embarked upon this human dietary intervention studies where we had 2 sets of participants, one group given a high fiber diet and one group given a high fermented food diet. We've heard throughout the day has sort of these diets having modulatory effects on the microbiome and sort of how, and some host readouts as well. And then we did complex microbiome and immune system profiling across a series of assays over time, looking sort of over the course of weeks at this interventions. And as I mentioned, I won't go into, I'll sort of give some highlights from this. If you're interested in this, this is published work that you can look at further. So just to start to look at what types of responses we saw both in the microbiome and the immune system, we saw 2 classes of responses. The first I'll talk about is the response to fermented food. And here what we saw was sort of globally, though across, though still substantial variation in this, we saw some global trends both in the microbiome and the immune system. One that I'll highlight here on the microbiome side was this increase in microbial diversity as measured by sort of the number of observed amplicon sequence variants, or ASVs, over time across the cohort. This was something that we could observe across this group. And what's paired with this was when we looked at the immune system data, particularly looking at inflammation markers, we can look both in our cytokine data over time, decreases in many of our measured inflammatory cytokines, as well as decreased level of sort of steady state activation across a series of different immune cells in sort of phosphorylated proteins. And so in this group, we saw these cohort-wide changes, both in an increase in microbial diversity with a decrease in inflammatory markers, again, sort of taken in aggregate. We can contrast this with what we saw in the high fiber diet group. And this was more of a complicated picture rather than a cohort-wide response. And so we did here is first we extracted our immune features, both coming from our O-link data to get serum cytokines, as I showed in the previous slide, and then metrics of cell frequency and signaling coming from CyTOF, as well as signaling capacity coming from an additional assay there. And we started to look at what were the just general responses if we look from baseline to end of intervention, how does this look across our cohort? And instead of sort of seeing this consistent pattern like in the fermented food, we started to see a series of subgroups tend to emerge, at least in the immune response, which we, again, kind of gave gross labels of sort of putative high inflammation group versus a low inflammation group 1 or low inflammation group 2, purely just on sort of how many of our immune activation markers go up or go down. Then we looked sort of at these groups, do these have any bearings on what we can see in our microbiome data? And what we see is that we can relate both to features at baseline and in responses. So at baselines or prior to intervention, what we saw is that the high inflammation group, the group that actually seemed to increase in response to fermented food, actually started with lower levels of microbiome diversity as compared to the low inflammation groups. We also saw differences in specific microbial taxa in terms of their responsiveness over time that tracked with whether we were getting the increase or decrease in microbial diversity. So I know I went through that a bit quickly, but what I wanted to sort of emphasize from this fact is that we did see sort of changes that were potentially in concert in the microbiome and the immune system, though really the focus was sort of looking piece by piece and seeing sort of what happens in this aspect of the microbiome or this aspect of the immune system and trying to find some relationships there. But we could look at this more directly, this idea of can we use this data set more broadly to infer sort of fundamental relationships and interactions between the microbiome and the immune system. So in this paper, we only sort of touched on this by doing sort of what I mentioned before, which is sort of where I was at when I was doing this work as a postdoc of let's do the easy thing of pairwise correlation. So if we take each of our data modalities on the immune side or the microbiome side and just looked at the number of significant pairwise correlations, sort of normalized by the number of features, we can start to see that there are certain modalities that had that we were detected certain relationships. Sort of the highest among them were relationships between immune cell frequency and levels of short chain fatty acids. And then also sort of a level of host proteomic technique with the level of Kzyme, so the degree of carbohydrate degrading machinery detected in the microbiome. We can drill down into that one and say, okay, what is the nature of this relationship? And specifically when we have something like host proteins, what is the actual signal that's coming out here in terms of the relationship that we're detecting? When we do this, we actually see that the sort of top relationship there is coming from an inverse relationship between sort of markers of inflammation and the number of Kzymes, or sort of the carbohydrate capacity of the microbiome. And so, like I said, this is something that just scratched the surface of how we can sort of put these data sets together, and something that I really wanted to tackle more explicitly in the work that we're now pursuing as a lab in collaboration with others. So I'd like to spend the remainder of the time talking about a collaboration that was actually started by Joel Babdor, who was a postdoc here in Matt Spitzer's group and now has his own group at University of Pennsylvania. This is led though together with Joel, of course, and then Ravi Patel, a senior data scientist in my group, and then us, of course, together with Matt. And this was this effort, sort of a Herculean effort from which we sort of play more of a data part in doing so, this immunomicrobiome project. And what was very exciting to me about this project was sort of the possibility to start to tackle data integration in the way that I'd hoped. And so the sort of background and context for this study here was that we had 110 healthy individuals that were sampled from their blood, stool, and urine. There may be folks in this room who donated to this project, for which we very much thank you. We have a sort of high enrichment of sort of the UCSF BCMM community. So there's that sort of covariate in the data to account for. But really the idea was to be able to capture sort of several aspects of the immune system, several aspects of the microbiome, and then the metabolic output of each, alongside sort of chronicling elements of lifestyle, like diet, sleep, different elements of activation, physical activity, vaccination, and sort of other parameters. And the goal here was sort of first and foremost to kind of create a reference of the healthy immune system, microbiome, and metabolome that could really sort of anchor also a lot of the studies happening both in the ImmunoX program and the BCMM program. Sort of can be very useful as we sort of look at sort of different more states of pathology. But then also, as something that I think I've made a point of, start to try to quantify this shared variation and start to infer the relationships across these two systems. And so what does this look like specifically? So from sort of these biospecimens that we collected, we had a series of assays of which we've run that are summarized here. And from each of those assays, we extract our feature sets sort of based on kind of best practices along each data set, such that we are going to then use as inputs to our integrative analysis approach. I do want to take a minute to talk about, before I sort of yada that into our integrative analysis, of sort of what it actually really took to be able to generate all of this data, and everything from sort of collection to getting to that nice cleaned up feature set that we can then do integrative analysis on. This is really something that where we're able to very much leverage the CoLab system. And so Margot mentioned in my introduction that as part of my research program, I lead the data science CoLab. And so for those of you who aren't familiar with what the CoLabs are, these are a series of labs that have come together with complementary expertise to be able to take on projects that would be very challenging for any one lab. And so is sort of some depiction of that here, but just to sort of highlight what this means kind of in practice. Sort of on the data collection and generation side, this is very much done from the disease to biology and genomics collabs with our collaborators, Alexi Combs and Brittany Davidson, shown here. A lot of the metabolomics work was done by Mariah Sandy, who was leading the quantitative metabolomics collab plugin. And then many of our data processing pipelines and feature extraction was done leveraging pipelines within the data science collab. And this was also done with additional support from the BioHub and sort of funding from BCMM and ImmunoX. So all of those forces came together such that we could do this study. And I just wanted to take a moment, one, to acknowledge that, and two, to sort of demonstrate the power of bringing this group together in team science. So digression on that over, but happy to also answer questions about the collabs model in future discussions, in addition to the research findings that I'll be discussing today. Okay, so all this group did all of this work. We have our set of features. We've been building up to doing integrative analysis. Here we go. So the approach that we decided to use here was multi-omic factor analysis, or MOFA, to be able to identify this shared variation across the microbiome and the immune system. So for those of you not familiar with MOFA, this is a joint dimensionality reduction technique for integrative analysis. And so what's really powerful here is we can take several different data modalities. We'll have sample by feature matrices for each of those modalities. And then the algorithm then will do matrix decomposition such that you can derive a set of factors that are going to be combinations of your features coming from various modalities in a mixed way, such that in the end, you both have your set of factor scores or levels for any given of your samples. And then you also have your sort of feature weights coming from each of those factors that you can then take a look at to see sort of which features from which modalities are contributing to each factor. So you can both get a measure of these factors for the samples that you have, but then also you can understand what those factors are and see sort of where that underlying signal is coming from. So we ran MOFA, and from this, I will just show you the top 5 factors we have and that we found and that we followed up on for analysis. So these are the top 5 factors here shown as where the sort of percent variance in that factor is coming from in terms of which data set. And what I'm showing here is sort of the bluish hues are our immune factors. So that's coming from our sort of frequency data and our single cell gene expression data. And then we have our series of microbiome factors. So the sort of composition and pathways of the microbiome, as well as the metabolic output there. And what I hope you can start to notice is, one, what's exciting is sort of getting contribution from various these modalities, but we were largely comparing sort of factor 1 and factor 3. What struck us about factor 1 versus factor 3 is you can see that factor 1 has a pretty sizable sort of immune contribution, whereas fairly minimal, relatively microbiome contribution. As compared to factor 3, you're really sort of seeing strong representation from both. Another way to be able to look at this is here. This is PCA plots of microbial composition colored by factor score, and we're in our sort of microbiome and immune factor, factor 3. You can sort of see this split by factor score across this microbiome PCA as compared to this sort of immune dominated. I sort of, for the sake of simplicity, call it microbiome independent, though not completely independent, feature in factor 1. And so from this, because we're really interested in capturing microbiome and immune shared variation, our focus is on factor 3, but sort of keeping factor 1 in mind as sort of a point of comparison for us to sort of understand. So now we can look under the hood at our immune and microbiome factor, see what's going on, see what features are really contributing to this factor score. And for those individuals, that may be a higher level of this immune and microbiome factor. So first, we can look on the microbiome side. This is looking at how all of the features associate with factor 3. And we can look first at the stool, simultaneously at the stool metabolites, as well as the data deriving from metagenomics. Here, this is colored by some of the specific pathways and their related metabolites. And what we see is a positive association with some of us sort of key players that we think of are in the microbiome. So we see positive association with polyamines, with short chain fatty acids, really sort of enriched in this factor. then we can pair that with taking a look at what's happening on the immune side and the immune contribution. And I'll sort of look at this in a couple different ways. So first we can look at immune cell frequencies. This is a scaffold map where you can just look at sort of positive and negative association of immune cell clusters that sort of cluster around landmarks of known cell types. We see positive association with several monocyte subsets, a bit of a negative association with some memory T cell phenotypes, but mostly sort of a positive association that we can then also look at. We also see positive association with a lot of immune system activation markers such as HLA-DR and others. We decided to then delve into more of the gene expression data to see which pathways may be enriched across these cell types. And when we do this, the sort of overarching factors that really start to kind of scream out at us from gene set enrichment analysis are interferon pathways, or both interferon alpha and interferon gamma. So this is really interesting. We got to this point where we see sort of higher microbiome activity, higher sort of interferon activity, all kind of contained in this factor. But we wanted to understand what is that nature of that immune activation? What does this mean? What does this look like? What was particularly complex or perplexing to us was when we, now if you remember factor one, the sort of microbiome more independent one, we also see some degree of activation of the interferon pathways. So at the top is factor 3 and below is factor 1. And so this is particularly important for us to disentangle which element of this sort of interferon level is related or sort of coincident with the microbiome activation or activity rather, as compared to sort of this more microbiome independent piece. So what's great is that we're not the first group to be looking at interferon. And we could take a series from the literature of more specific interferon signatures and see how those play out in our factors as we compare the two. And so more specifically, we can compare what has been described as a tonic interferon gene signature as compared to more of an acute interferon gene signature. So this is actually derived from the mouse to be able to get genes that are associated more with sort of a baseline steady state rather than an active infection or sort of active response likely from an infection. And so if we take a look at that tonic interferon gene signature, that one is specifically activated or associated rather with factor 3 as compared to factor 1. And I don't show it here, but neither of which are enriched for the acute interferon signature. So it seems like this is very much specific to this tonic interferon signature, and it's specific to factor 3, our microbiome and immune factor. And so what could this mean? We still have further validation work to do, but what this could point to is how this signature is actually described in the mouse has actually been the mode of which that commensals sort of toggle and calibrate the interferon response to better calibrate response to viral infection. So it may be that we're detecting kind of this shared signal, but in this case in humans, of potentially sort of commensal activity sort of shaping that response. So just to wrap up with the end of my time, hopefully I've introduced a framework for starting to interrogate this sort of covariation between the immune system and the microbiome. I talked to you about sort of how we sort of looked at these systems in the context of dietary intervention, and then how we looked at this more specifically in steady state and what we're starting to detect in terms of shared variation across the microbiome and the immune system. With that, I'd like to thank my lab as well as sort of the leads in the immune microbiome team, including Joelle, Ravi, and Matt, and also thank my Stanford collaborators and all those across CoLabs and ImmunoX. And I'd love to take questions at their time. Thanks.
Hey, Gabby, really enjoyed that. Were there viral sequences that were included in the metagenomic data, in your input data for the factor identification?
So we were looking at that a bit. We actually more our approach that we've been running IDSeq to start to take a look at the, and it actually does look like, so we do see some association of potentially some viral signatures with factor 3 in some of our. We're worried about just sort of like the full kind of integrity of that signal because that's just sort of an approach we haven't done as much. So we haven't been including that yet, but we see inklings of there being sort of increased viral there too.
So nice talk, Gabby. So if I understood correctly, the samples that you analyze come from healthy individuals. So you're not associating these factors to any particular phenotype. It's just internal co-regulation.
Exactly. Steady state. Yeah, what we can infer from that.
Gabby, I guess I have a question. The MOFA model, do you see, how would that be applied to other kind of paired systems, you know, outside of the immune system that you, immune microbiome that you specifically focused on?
Yeah, so it's nice. It's actually a really pretty general technique, and you can sort of on any type of multimodal data you can use it for. There are some caveats around being sort of careful around comparable dimensionality and sort of things like that, but you know, I don't think we've seen it applied quite this way for the set of modalities, but you know, you see it in sort of even just looking across immune modalities, and it could work in lots of different systems.
Any other questions? Okay.
Thank you very much.
So our next speaker is going to be Dr. Javier Ceja Navarro. Javier is an associate professor in the Department of Biological Sciences at Northern Arizona University. He received his PhD in biotechnology from the Center of Research and Advanced Studies, National Polytechnic Institute, Mexico. His research is focused on the study of multitrophic interactions in ecosystems, such as the digestive tract of insects in soil. Specifically focused topics of his research include the study of arthropods as microbial bioreactors, coevolution of insect digestive tract physical structures, and microbial function for the transformation of recalcitrant molecules such as lignocellulose, and environmental engineering and regulation of ecosystem services driven by the multitrophic interactions among the members of the food web of complex ecosystems, their contribution to ecosystem functions, and their responses to environmental changes.
All that. I want to thank. So thank you so much for, for the introduction. Let me see.
And Javier will probably mention it. When we invited him to originally present, he was in the Bay Area, which he is no longer. So he'll speak to that a little bit.
Oh, there we go.
Yeah.
Okay. Well, first of all, thank you for the invitation and for the lovely introduction. I'm Javier Navarro, and my laboratory is the Laboratory of Microbial Trophic Interactions. And as you heard, I work in systems such as soil and the microbiome associated with the gut of insects. And there we try to have an overview in which we try to integrate all the members of a microbiome, from the viruses to the bacteria to the fungi and the protists. But today, I want to tell you a couple of stories about the insects that I studied while I was here at the Bay Area, and will give you some glimpses of the work that I still carry out with me in Northern Arizona University. So first, I'm going to introduce you to my first character of this story. This is the Pasalid beetle. You can see the insect there. And specifically, we were working on characterizing the compartmentalization of microbial function for lignocellulose degradation in the beetle gut. I'll explain more about that. So about the insect, well, the Pasalid that I studied is native to the United States. There are around 960 species that are described around the world. And well, they are very interesting creatures because they communicate with each other by acoustic signals and chemical signals. They are also subsocial, and all stages feed on frass, which is a way in which they are transferring the microbiome to one another and members of the family. And only the adults process the wood or wood biomass because they are the ones that have the very strong mandibles that can actually chomp, oak wood or other types of hardwoods. And this is what makes this insect very special because it's a machine of, wood degradation. In 1 year, a single beetle can consume 1 kilogram of wood. So you can imagine how important they can be for ecosystem services in the forest. Thinking about or talking more about the cool things about this beetle, Well, let me show you the digestive tract of the animal. Just like our digestive system, the digestive system of the beetle has morphological differentiations that I'm showing you here. Here you can see the 4 different areas. We have the foregut, the midgut, the anterior hindgut, and posterior hindgut, and I'm also showing the tracheal tubes. So when you leave the beetle's beak, okay, the digestive system looks like this, like what I have here within the beetle. But when you are dissecting them and you extend the gut, you will get a long strand of around 12 centimeters. So it's a big piece of digestive system. So I need to give you a full disclosure. I am a trained chemical engineer. So when I was invited to go to the Berkeley lab to work with this beetle inside its microbiome, well, the first thing that I thought was, this is a bioreactor, and that's how I'm going to treat it. So there you have my representation of the beetle. And the question was, or the question that we were pursuing was, is the morphological compartmentalization of the gut an adaptation to segregate microbial function, in specific to transform lignocellulose? You know, this animal is consuming lignin, cellulose, and hemicellulose when it's feeding on wood, and it's pretty much surviving on this low nutrient diet. So the question was around those lines. How is the beetle managing to do this? So of course, the first test that we attempted to do, or that we did, was to actually figure out whether the beetle was transforming this carbon as the wood was moving to the digestive system of the animal. And there is where I had my free experience collecting frass of insects. That's how we call the poop of insects. And what we did is that we fed the beetles with oak wood. And then hour by hour, I was monitoring the beetles to remove any pieces of frass that they were producing, because they tend to eat it. So I just wanted to force them to keep eating the hardwood. So once we collected our sample, we applied bulk chemistry, and I'm going to show you the results here, and also thermochemolysis. So on the first part of the graph where you see bulk chemistry, we are comparing total carbon and total nitrogen when we have wood and frass. So wood that has been processed. And as you can see, we have there significant differences in total carbon, but also we have a significant increase of total nitrogen in the frass, which makes you think, you know, you have here an insect that in the forest is chewing plant material and is releasing all this wonderful nitrogen for the ecosystem. I mean, we know that termites can do that as well. But we also wanted to know, you know, is lignin being transformed? You know, lignin, I'm sure that you know this, is very hard to transform. Even in the biofuel industry, we still don't know how to process lignin. So we're wondering, is this beetle able to do this degradation of lignin? And that's the plot that I'm showing you there. We applied 13C thermochemolysis to characterize the differences in acid to aldehyde ratio between wood and poop. And as you can see, well, we have a lot of acid compounds that are released or that were detected in the frass, but not that much in the fed wood. So here we have this beautiful system that is able to transform carbon, that apparently is able to transform lignin, but also that enriches possibly the environment with nitrogen. So we wanted to know, how is this happening? How is the beetle doing this? So we decided to deploy multiomics, and specific metagenomics, and also proteomics and metabolomics. You know, and this was a wonderful thing about working at the Berkeley lab, and all the collaborations that we can have that came to be very easily. So I'm going to show you the results in a very, that's not one of my favorite plots, the one that I'm about to show you, but please bear with me for a moment. So here, what we can see is the average coverage of putative microbial lignin degrading genes, and also those genes that are related to hemicellulose degradation. And you have them there for the different regions of the beetle gut. Initially, when we did analysis of our metagenome, we were gene-centric. So we're focusing on any of the genes that we could detect in the metagenome that were related to the lignocellulose transformation. So what we found was that specifically those genes for lignin, cellulose, and hemicellulose transformation were highly abundant in what we call the midgut and then posterior hindgut, you know, the blue area and then the last yellowish area in the beetle gut. We applied metatranscriptomics, and then we found a similar pattern of distribution, more markedly for those genes or those proteins involved in lignin degradation. As you can see, they are highly abundant in the midgut. So with this evidence, well, we decided to also apply metagenome or to identify these metagenome assembled genomes to identify who is performing these tasks among bacteria. And therefore, I want to show you some of the MACs that we reconstructed that carry the genes involved in these processes. So with this in mind, and going back to my model of the bioreactor, well, now we can see that depolymerization is happening in these two areas that I'm showing you there. So at the beginning in the midgut, but also by the end in the posterior hindgut. So that was a cool thing for us to find. And the question, well, there was why? Why would the beetle apply a lot more energy to its microbiome or request more of its microbiome to continue the degradation of lignocellulose also in the posterior hindgut, when the material is about to be released in the form of poop, right? We will get to that in a moment. So here we have depolymerization. As you may know, when you degrade cellulose and hemicellulose, you release glucose and xylose. So the question now was, what is happening with these products of depolymerization in the beetle gut? So we continue with our metagenomic and metaproteomic analysis. And we elucidated the main pathways involved in the transformation of these compounds, which were related to fermentation. Here you can see again the distribution of the different pathways in the different parts of the beetle gut. And we elucidated there that through fermentation, the beetle could be producing lactate, formate, acetate, or even ethanol. You know, one important thing here is the production of acetate, because acetate is the energy currency for beetles and for the microbial communities. So we found that not only acetate was being produced by fermentation, but also by homocytogenesis, both the carbonyl and methyl branches. We also identified key players in these processes in the beetle gut. Well, here, now we have depolymerization in these two areas, but then we have fermentation in the anterior hindgut happening in the beetle gut. So a key thing for fermentation to happen is that you need to have anaerobic conditions, correct? So what we did was to use these microelectrode sensors. You know, these are micron scale sensors that have a tip of 10 microns. So there I was dissecting beetles, opening the beetles, and then measuring the gradients with my microelectrodes, just going through the surface of the gut and then moving all the way through the gut. Here are the profiles that we identified at the anterior hindgut. So as you can see here, when you're in the surface of the beetle gut, I cannot find the cursor, and I don't know if I can move over there, otherwise I will go and point. Oh, can I do it? Okay. So when you are at the very surface of the beetle gut, you see that we have certain levels of oxygen around 20 micromole per liter. But as you start moving through the gut, you start to see that there is a depletion of oxygen and then the formation of a totally anaerobic core. So there we can say that our bioreactor, in specific, the anterior hindgut, has the conditions for fermentation. But not only that, you know, we detected these gradients in the different parts of the beetle gut with more or less distribution of oxygen and anaerobiosis within the gut. So we take this as an indication that maybe there is the occurrence of these processes, both aerobic and anaerobic within the beetle gut. I think I'm good on time. So we then decided to apply what I call the ultimate test. You know, can we actually detect any of these metabolites that we reconstructed with metagenome and metaproteome? So we fed our beetles a group with oak wood, and another one was tortured to consume chemically pure cellulose, hemicellulose, and lignin, you know, the one that you buy from Sigma. So they were fed with this. I was there again, waiting for them to produce their frass, collected the frass, and then apply NMR. I want to show you the results of this analysis. You can see metabolite concentration, different metabolites that were detected. We can see that for frass from beetles that were fed with wood diet and others with artificial diet. And you can see there that the frass is actually highly enriched with all these metabolites: acetate, ethanol, formate, lactate, propionate. So that made us think differently about frass and about the poop that is produced by beetles or released by beetles. If you recall at the beginning of the presentation, I mentioned that it's only the adults that are able to feed on the hardwoods, because the babies, the larvae, they don't have the developed mandibles. But another thing that is very interesting about these big beetles is that you can see them. You can follow them when they are in their colonies. And there is certain behavior there that many have recorded. And that is that the larvae spend their whole time just following the adults. So imagine you and your little cousin just following you, not for the same reasons. But what they are doing is that these larvae are following the adults to get access to this nutrient-rich grass. Researchers have tried to grow these larvae with different substrates, and they simply cannot develop. So there you go. So what we have found here is that the establishment of microbial partners require the development of specific habitats within the gut, right? And that anatomical development and microbial functional assembly are allowing this beetle to survive in this low nutrient diet, but also to allow the whole colony to survive in recalcitrant wood. Now, if you think that I'm done, you are wrong, because I want to tell you about another beetle. You know, it's about the same idea, about how we are working on characterizing how microbes are helping insects to pretty much be these powerhouses that they are. So here I want to introduce you to another insect that is empowered by microbes. This is the coffee berry borer. Has anybody heard about this beetle that is not soup? Yes. Okay, I'll tell you about this beetle. This beetle is the most important insect pest of coffee. Pretty much can be found in all coffee producing countries. When a female berry borer flies and finds a coffee cherry, it will bore, create a hole, get into the seeds, lay eggs, and then everybody starts to have a feast on this precious coffee bean. And there you can see what happens with the coffee bean. So this is a big problem because in some countries, the insect can generate losses of around 80% of crop production. So it's a big problem. Add that to other pathogens. So you see we may have a big problem in coffee production in everywhere pretty much. But another thing that makes this super interesting is that this is the only insect that is completing its whole life cycle inside of the coffee bean. We know that caffeine is highly concentrated in the coffee bean, right? But I'm sure that you know as well, the caffeine is produced by plants as a mechanism of defense against herbivory. So that's supposed to keep insects away from eating the plant. Well, nothing is happening to the coffee berry borer. And here I'm depicting the different problems that one can have when consuming a lot of caffeine, you know, cell wall damage, impairment of protein traffic, and so on. And here's the interesting thing, you know, if you calculate the amount of caffeine that an insect is consuming through 10 hours of just feeding on coffee, you will figure out that the insect is consuming the caffeine that is contained around 500 shots of espresso. So imagine you yourself consuming that. So the question here is, who is helping the beetle? Is it the microbiome maybe helping the beetle to degrade the caffeine? So the first thing that we did was to characterize the microbiome of insects from different coffee producing countries. You know, in that very colorful and busy image, you can see a distribution of the different microbes that we detected there. Of course, there is a lot of Pseudomonas, there is a lot of Enterobacteria, even gram-positive bacteria. The idea started in general just as a study of biogeography. So we found that yes, with analysis of covariance, you can see that community varies and is explained by location. But there is a core microbiome that is represented by many of the organisms that I'm showing you here, right? Well, you know, what started just as this biogeography study at the beginning of my postdoc, well, then got turned into a nightmare. Because I was tasked, no, I'm kidding. It was a beautiful project. I was tasked to then, so please, no. I was then tasked to isolate the caffeine degrading bacteria. You know, and I said, yeah, I can do it. Absolutely, I can do it. No, the beetle is only 2 millimeter size, so I'll manage. So there I was dissecting the beetles. And here you can see that after a lot of training, well, I managed to dissect the guts, which is what you can see here. You have the crop and all the way to the anal cavity there. So once we dissected those, we started to grow microbes in caffeine plates. These agar plates only contain pure caffeine. There was nothing else in there. And I want to show you there the distribution of microbes in these caffeine grading plates that go from low concentration to high concentration. Now, one cool thing about this project is by the time when we were isolating, there were maybe 1 or 2 bacteria reported as caffeine degrading. And here, well, we were able to isolate all the ones that I'm showing you there. But then I had this very good supervisor that asked me, well, even if they are able to grow in this caffeine, does it mean that they are actually degrading the caffeine within the beetle? So you need to figure out. I will show you what we did after. We extracted DNA and RNA from beetles from the field, synthesized cDNA, and looked for one gene, one reported gene for caffeine degradation. It's called the NDMA. And I'll show you the result in this form just because I love it and because I guess I'm old-fashioned. Actually, my first sequencing was also Sanger, just to say, and I love electrophoresis gels. So I want to show you the results here. It was qPCR, but I still run the gel. So there you can see that we could detect the expression of the gene in the cDNA. Our RNA was not contaminated. It was very clean. And of course, we detected in the DNA, you have the positive, etc. So that was the idea. Well, it's active within the beetle. So I cloned the genes that we just amplified and compare it against the genes of all my isolates. These come from the beetle, but I also amplified them from my isolates, the caffeine degrading bacteria. And here you can see that they are forming a cluster from isolated bacteria and then the ones that we amplified in the beetle. So then again, I'm super happy with these results. And a colleague asked me, well, why don't you get rid of the bacteria and see what happens? Right? And very, very easy. But it was a great suggestion because then we moved to manipulating the microbiome of this beetle. So specifically, what we did is that we took beetles with their normal microbiome, placed them in these 96 well plates that contain diet, a coffee smoothie that we gave to these beetles with fluorescent beads. So we put the beetles there to eat, take them out, move them to a new little plate, let them there to defecate, remove them, and then I went back to look for the spheres, because if the spheres were there, maybe there was frass there, right? Maybe there was poo. Am I trying to collect the poo or something that small? The next test was to treat the beetles with a cocktail of antibiotics, and then to regular diet, collect frass, measure caffeine. To then finally, the same, beetles with antibiotics, but then they were inoculated with these caffeine degrading bacteria to then see if inoculation did anything to them. I want to show the results here. You can see that in our control, which is these beetles with the regular microbiome, when we measure the caffeine in their frass, well, there is very little caffeine released, at least as I said it by GCMS. When we apply FTIR, we couldn't detect a thing. When we treat them with antibiotics, you can see the levels of caffeine that we are detecting. These levels of caffeine are very similar to the levels of caffeine in the diet that we're providing them. So pretty much the caffeine we're just going through. And finally, when we reinoculate, you can see again that there is a reduction in the caffeine content in the gut. So that was enough evidence for us to show that this was a process that was mediated by the microbiome. Later, we also created a draft genome for the beetle, and immediately we started to look for the genes involved in caffeine degradation, at least the ones that we know, and they weren't there. There have been events of horizontal gene transfer or other genes in the coffee beetle, but not for caffeine degradation. So what's going on? I missed that part. So what's going on now? So as you know, we are what we eat, right? So now we are studying how caffeine concentration affects the microbiome of the coffee berry border. And for this, well, we're collecting beetles that are found infesting Arabica coffee and Robusta coffee. These are the main 2 coffee varieties or species that are grown in all coffee producing countries. The big difference here is that Arabica has lower caffeine than Robusta. So we collected beetles from the field in Chiapas specifically, but now we went to collect hundreds and hundreds of insects from each of these. We did microbiome analysis, and we saw that there was a very strong effect, at least on coffee species, on microbiome composition. From here, we reconstructed networks. That was the key thing for us. We wanted to get hundreds of insects to be able to reconstruct a network and identify what we will call potential keystone taxa or keystone elements in the network. which I'm showing you here in color. Keystone nodes will be defined are those members of a network that when removed from the network will create a collapse in the whole network that you have reconstructed. Specifically here, the keystone nodes that we have identified are Pseudomonas, Stenotrophomonas, and Microbacterium, which are all caffeine-degrading bacteria. So now, well, I'm working at NAU, as you heard, and we are working in the isolation of phages and the expression of taylorsins to manipulate the viral microbiome. For this, I'm working with my colleague Vivek, and what I'm going to show you here some of the results of the genome analysis of our isolates. We can then identify some clusters that resemble those of taylorsins in Pseudomonas. But another key element for us is identifying microbial interactions. I think it's important for everybody working in host microbe associations, and that's something that we are trying to pursue here. And I'm going to show you some of the results that I have generated from a different study. In this case, is a microbial community that was adapted to grow in sorghum. And then after growing up sorghum or enriching for these microbes, they grew in sorghum with high lignin and sorghum with low lignin. So again, it's a bioreactor. So what we have done here to elucidate potential interactions between microbes is to create metagenome-based networks, which is what I'm showing here. So we take a whole network, a lot of replication, reconstruct our networks, and over this, we then overlay, oh, Jesus, all right, there, our metatranscriptome data. What I didn't mention is that we follow the metatranscriptome in these bioreactors over time, which is what I'm showing there. So then we look for signals for the areas that are activated among microbes and look for the expression of genes such as secretion system or the activation of genes involved in competition such as antibiotic resistant genes or the activation of transporters. And based on that, we, well, created or established potential interactions among microbes. Was published last year. I'm going to finish here again with the coffee beetle to say that the insect has many bacterial members that can subsist on caffeine as their sole source of carbon and nitrogen. And well, that these microbes are responsible for caffeine degradation, and well, that caffeine, a toxic alkaloid, mediates microbiome composition in the beetle gut and makes it to be the beautiful creature that it is. So I'm going to finalize this just to thanking everybody that worked in these different projects at the Berkeley Lab, Livermore, UC Berkeley, PNNL, Owen, and Ulash, that taught me a lot of what I know and what I do now. All the different undergraduate students that I had at the Berkeley Lab, and now, well, the new members of my laboratory. So thank you so much.
Javier, I actually have a question for the first beetle. Apologies for the name. Did you say that the adult beetle also consumes its own frass?
They do.
So what do you think of the selective pressure evolutionary advantage of that, given it's in not a resource limited, environment, has access to plenty of wood?
I will say that it's a way possibly to maintain their microbiome, so they continue to be reinoculated, and also to acquire some of the same nitrogen that they are producing for the environment. So that's one of the ideas that we have.
Yeah. Really interesting. Maria first.
Wonderful talk. So my question is about the coffee borer, and I wondered if its presence affected the caffeine level in the coffee cherry. Is there any compensatory production of caffeine, or does it go down?
I don't know. We haven't tested it. Here's the thing, the thing is that when farmers detect any cherry that has even signals of being affected by the insect, they pretty much burn it. They are trying to get rid of everything, but that's a good question. I should check on that. Thank you. Yeah.
Javier, do you have a sense whether the ecosystem function of those caffeine degraders is purely detoxification, or are they providing some other metabolite to the beetle that allows it to grow? Or
When we did the genome analysis of the beetles, we can see that they have genes for participation in fermentation and many other processes within the gut. So we believe that it's not only that, so there is more that is going on there. Another thing that I didn't mention is that we also characterize the gradients of oxygen and hydrogen in these tiny beetles using the microsensors. So we see there that there are the conditions for, again, what I'm mentioning, fermentation and even different pathways. So there is more going on there.
So if you feed these guys C13 labeled caffeine, do you see that label move into the beetle tissue? Or
we need to do that. Many years ago, we tried, and we were working with our colleagues in Livermore. They gave us the labeled caffeine, but it was too little of a sample for them to do the analysis back in the day. So we'll see. I'm sure that we can try again. Yeah. Yes, absolutely. Yeah. Thank you.
That was great. Thanks. I was just curious if there's any evidence for caffeine degradation in the environment outside the beetle gut, so in rotting coffee beans, or is there maybe exchange of caffeine degradation in genes?
There may be, I think, because remember that I mentioned that we were isolating these caffeine degrading bacteria. There were a couple that were reported by that time. They were isolated actually from soil of coffee plantations. So it is possible, that the beetle is carrying this microbiome from different areas. One thing that is important to consider is that when there is no coffee, there are no cherries for the insects to infest, they go on the ground. So there is a good ecological story there that maybe we can pursue. Yeah.
Cool, thanks.
Mm-hmm.
Yeah, great question, or great talk. Kind of building on the question about toxicity, when you depleted the microbiome with antibiotics, did you see deleterious effects on the beetles?
Okay, not back in the day. Here's one thing that I need to tell you. When we did, and we reported this paper, the diet that we used was the diet that, other scientists that work with this beetle were using. This diet has 1/10 of the caffeine that is found in the coffee beans. So we try to measure if that will be enough to create a negative effect on the beetles, right? And we see, some of them weren't really growing that well or reproducing, but nothing that we could call significant. Now that I am an NAU and can work again with the beetle, we are creating formulations of this diet that goes all the way to 40 milligrams per gram of caffeine, which is twice what you will find in the environment. And we can see that they may reproduce, but then the larvae die. Okay. So they don't reach full development. So we are following those cool. Little pieces there. Yeah. Thank you so much. Yeah.
So our next speaker will be joining virtually. She is Dr. Angela Zakovich. She is an associate professor of Department of Nutrition at the University of California, Davis. She received her PhD in nutritional biology from the University of California, Berkeley, and her research is focused on the role of diet and nutrition in precision health. Please join me in welcoming Angela.
Okay, can you guys hear me okay now?
Yes, loud and clear, Angela.
Oh, wonderful. Okay. Thank you so much for inviting me to the conference, and I'm very sorry that I couldn't be there in person. So that was a really wonderful series of talks, and for now, we'll just kind of go back to humans. That was very cool. I had no idea beetles could be that interesting. Okay, so why is nutrition so complicated? Well, first of all, we know that there is this high level of inter-individual variability in response to diet and nutrition. But when you really start to think about it, it becomes almost sort of overwhelming to realize that we not only have, our genotype that drives things, the intrauterine signaling and early childhood influences that influence, the early, setup, but then there's also all these changes over time. And then there's also various cyclical things that can happen, diurnal cycles, monthly cycles, all these things that are constantly influencing how we respond to different foods. And so this is one of the things that's really challenging about studying diet and the gut microbiome. So here I'm just showing a very simple graph that really kind of shows this point that individual response is highly variable. And this is even in the simplest possible parameters. So this is just looking at plasma triglyceride concentrations in young, healthy, white male subjects in response to a lipid challenge, the same exact lipid challenge meal that they drank. And you can see this huge variation in the amount of triglyceride that shows up in the bloodstream. So this is just the simplest possible variable that you can look at in the simplest possible type of dietary challenge, and yet you still see this really high variation. So when it comes to the gut microbiome, of course, this gets even more complicated. I'm sure many of you have seen many of these figures. They're kind of classic figures at this point, showing that we have a core gut microbiome, that we all share certain microbes because, you know, the gut niche promotes that whereas functions are largely conserved, we do have a very high degree of inter-individual variability in the species that are actually there that confer these functions. And basically what this means is that there are many different kinds of healthy microbiomes. So it's not really that straightforward to say, here's the target healthy microbiome we should all be aspiring to, because there's so many different ways to have a healthy microbiome. The gut microbiome, of course, is involved in many aspects of health, and dysbiosis is associated with many different disease states. And we can certainly say that we know what an unhealthy microbiome looks like. So we know that an unhealthy microbiome has an increase in pathogenic microbes, a decrease in beneficial microbes, typically also a reduction in diversity. And so we kind of know that those are the general targets, right? To increase certain beneficial microbes and to decrease certain pathogenic microbes. But given this high inter-individual variability, how do we study the effects of diets on microbiome? Well, that's been really the focus of our lab, is to really figure out what is the right study design and what are the right approaches for studying how diets can affect the microbiome. And I'll try to convince you that the crossover study design and selection of the study population are really critical parameters. So choosing the study population is key because there are things that we can do by simply increasing sample size and then accounting for things statistically. That is true. However, if we really want to look at what is the effect of a particular dietary intervention on aspects of the gut microbiome, it's probably better to, in fact, try to sort of reduce some of the noise that contributes to changes in the microbiome so that we can actually see the signal of the diet itself. Many different factors contribute to fluctuations in the gut microbiome. Here are some of them. Everything from hydration status to menstrual cycle timing to various medications, to environment, like where you are, all can influence the microbiome. So if you don't try to account for some of these, it will be very difficult to discern the effect of the intervention from the fluctuations due to these other factors. So here's kind of an illustration of, this idea of what a nice signal would look like from the intervention when the signal from the intervention is higher than the noise from the other factors. And then here's kind of, an illustration of, unfortunately, what it looks like if you have too much noise in there and it really just masks the signal. And then the crossover study design is the other general approach that we use for really trying to see the signal very well. So the crossover study design is essentially, where you have each individual going through both dietary intervention arms with a washout period in between, and being randomized in terms of order to either start on treatment 1 and then go to treatment 2, or start on treatment 2 and then go on treatment 1. This approach helps account for the large inter-individual variability in both the background microbiome and also in the responsiveness to the intervention. And it increases statistical power. So each person acts as their own control, and you really don't need as large of a sample size because you can use a more powerful statistical approach, the linear mixed model, and you can see effects of even subtle interventions, right? So you don't have to have huge diet changes to be able to see changes in the microbiome. And then finally, another advantage is that this provides a path toward precision health, where we're not just looking for an average response, but actually looking for sources of individual variability. So the ideal study design, of course, would be the double-blinded, placebo-controlled, randomized order crossover design. And of course, just a quick note that the crossover design won't necessarily work in all situations. For example, if you have a population with a progressive disease, then it just won't work because there'll be at different stages of the disease at the two different start points. So how do we apply this crossover study design? We did this in the first case by trying to increase the abundance of beneficial microbes through diet. And we wanted to try something, you know, that is basically kind of a low bar and also that is very practical in nature, right? So most Americans are not meeting fiber recommendations. They're probably getting, you know, less than half of what is recommended. And there are unfortunately many barriers to dietary modification. So you have to have access to healthy foods. They're more expensive. You have to have the ability to prepare them. They go bad more quickly. There's various cultural, financial, you know, other barriers to saying, well, everybody just start eating more fruits and vegetables. So we wanted to see, is there some other approach? Oh, sorry, that's a little jumpy there. To do this. There are multiple lines of evidence suggesting that increasing the relative composition of certain beneficial microbes, like bifidobacteria, have beneficial health effects. So including improvements in gut permeability, anti-inflammatory effects, and then effects on metabolism, for example, reduction of cholesterol levels. So our hypothesis was very simple and straightforward. Could we just give a very simple to implement modest dose of dietary fiber? This is only 12 grams per day, designed to kind of get them close to the recommended amount. And with this simple intervention, it's just a powder you put into a glass of water, you drink it, you don't have to change anything else about your diet, you don't have to spend very much money or time. It's just a very simple intervention. Can this actually at least do something to help the gut microbiome? And in this case, we hypothesized that it would increase the abundance of bifidobacteria because of the contents. The prebiotic essentially had mostly inulin and some resistant starch. So the study population, we really took a long time to, you know, make sure that we had the right population to look at to, again, reduce that noise. So this was just healthy male and female young individuals with overweight BMI. Their daily fiber intake was to be below 15 grams per day. And we really needed to recruit people who had, you know, bowel movements with normal frequencies, because we actually experienced that from another study where people weren't able to produce a stool sample at the right time point because, you know, they couldn't do that. So we have lots of different exclusion criteria to make sure to control for things, even things like planned international travel. We learned that we really have to look for, because when people go, you know, travel to India or something like that, their microbiome completely changes. So you're going to really get used to seeing my little crossover study design figure here. Here's what it looks like. Essentially, people were randomized to either receive the fiber first or the placebo first. And this was a double-blinded, randomized order, placebo-controlled crossover study. And at both pre and post time points, we collected stool, diet information through 3-day diet records, as well as, you know, of course, plasma. And then what we introduced in this study was this idea of a patternized diet, because we had been aware of some work that had been published showing that, you know, you can have blooms in certain bacteria within 24 hours, and that you can have pretty drastic changes in gut microbiome just from going out to dinner to a restaurant where you don't normally eat, eating foods you don't normally eat. So we really wanted to stabilize the microbiome in the 3 days preceding the stool collection in that 24-hour window of the study visit. So we asked the study subjects to, on day -3 before each visit day, to eat a breakfast, lunch, and dinner, and then eat the exact same breakfast, lunch, and dinner, and breakfast, lunch, and dinner for the next 3 days. So we didn't want to artificially change their microbiome by giving them a standardized diet, because that would then, we would need to give them then time to adjust to the standardized microbiome. Instead, we just wanted them to eat their own foods, their own meals, but just make them really consistent for the 3 days leading up to the stool collection. Basically, these were generally healthy young individuals, normal glucose, normal lipids, so none of those things changed, and we really didn't expect them to. The participants' background diets did not change throughout the study. We really babysat them very much and made sure that they kind of continued eating the same things that they were eating at the start. And so the results show that just like we hypothesized, the amount of bifidobacteria increased, both at the genus level and also several of the species increased. And again, this was exactly what we expected because the prebiotic contain inulin, and of course, bifidobacteria are very good consumers of fructooligosaccharides. We then looked a little further since we did metagenomic sequencing and found that the prebiotic increased the abundance of several genes involved in short-chain fatty acid production, and that changes in the abundance of these short-chain fatty acid production genes were correlated with the change in bifidobacteria abundance. And then what we found that was kind of interesting that we didn't expect was that the change in bifidobacteria also correlated with changes in this choline dehydrogenase gene. And I'll come back to that in just a second. But first, just to show you, we also looked at the plasma metabolome. This was done through Metabolon. And what we found was that the concentrations of certain beneficial microbial metabolites like indole propionate increased after the prebiotic. TMAO, however, did not. And then another surprise was the decrease in choline itself and also in the long-chain acylcholines. So these acylcholines are actually modulators of cholinergic signaling. I really didn't know anything about them before this, but this was a really interesting outcome of this study. And it's an interesting question whether utilization of choline by bifidobacteria may redirect the choline from other microbes. So even without changing the background diet, this low dose of prebiotic, we were able to see an increase in beneficial bacteria without changing the overall microbiome. And we were able to change plasma metabolome. So there was an increase in beneficial microbial metabolites that was reflected by the change in this bifidobacterium abundance. So, you know, this careful study design can reveal changes even with really, really, really subtle interventions that normally might be missed because we've really reduced a lot of noise. So, but what about the decreasing of pathogenic bacteria and their metabolites through diet? So we've seen how we can increase beneficial bacteria, but what about decreasing the bad guys? So back to this question of choline and TMAO. So choline is converted in the gut by certain bacteria to TMA. I'm sure many of you have already seen this story. TMA is then absorbed and converted in the liver to TMAO. TMAO is this unfortunate metabolite that has been independently associated with prediction of cardiovascular disease. And this is even after adjusting for LDL cholesterol and many other cofactors. Choline is enriched in eggs. And so we were really curious what the effects of eggs might be. Just to show you real quick, this is a really interesting thing that was found 10 years ago, that the gut microbiota of omnivores, so Americans, typical Americans eating high animal food diets, actually produce quite a lot of TMA in the postprandial state. This is labeled choline that they gave, whereas the gut microbiota of plant eaters really don't produce any TMAO. But a really interesting point, again, is this really high inter-individual variability among the omnivores. So you could be an omnivore who basically produces nearly zero TMA in response to choline, and you could be an omnivore who produces quite high amounts of TMA in response to choline. So the question mark is, you know, what is really driving this? There's been several studies on this topic, and, you know, again, showing lots of really high inter-individual variation. And it's not really clear whether it's the long-term or background diet that is the main driver of TMAO production, or whether, you know, these short-term changes in substrate, so amount of choline that's coming in, can actually influence TMAO production. So we did another crossover study, and we gave postmenopausal, mildly hypercholesterolemic overweight women either 2 whole eggs for breakfast every day or egg whites. So this was a controlled feeding study where we gave them the breakfasts, but all of the rest of their food was just the normal food that they ate. And we monitored their diet to make sure that the background diet stayed the same. So this resulted in about 250 milligrams per day of extra choline from the eggs on the egg arm. And basically what we found was that choline increased, but basically TMAO did not. We found that there was basically no change whatsoever in microbiome diversity, no change in overall microbiome composition, and there were no changes in bacterial taxa that were previously shown to be involved in the conversion of choline to TMA. So here's a couple of them shown here. And basically it looks like the effects on the microbiome were null. So without changing the background diet, just changing breakfast to contain 2 whole eggs versus egg whites did not alter the gut microbiome composition. And despite the fact that it increased the amount of choline that was coming in, and you could see that very clearly in the increase in choline in the blood, the microbial metabolite TMAO did not increase. So in this case, what we can say is that the background microbiome, rather than the dietary changes in substrate, are really what are determining the amount of TMAO in the circulation. So we can say that because all the other factors influencing the FMO enzymes were essentially being held constant because everything else about this was controlled and individually controlled. So the next question we wanted to ask was, can we use short-term dietary challenge to discover whose microbiome is susceptible to, these bad effects from certain nutrients that could be bad for you, but maybe aren't bad for everybody? And here's where I would like to tell a story about another bad guy. So here it's Bilophila wadsworthia. And this is, I'm sure many of you are familiar, work by Suzanne Devkota, who showed that saturated fat diets can cause a bloom in Bilophila wadsworthia. And this bloom in Bilophila wadsworthia after high saturated fat feeding was associated with a really significant increase in colitis in susceptible, these IL-10 knockout mice. Interestingly, this is from another study, Bilophila wadsworthia can actually induce inflammation without changing the overall structure of the gut microbiome. So this is one of these microbes where you just kind of insert some of this bad guy, and without any changes to the rest of the microbiome, it has direct negative effects. Are we all at equal risk? After looking at this, you might think to yourself, oh my God, I should not eat any saturated fat, no more cream for me. But is this true? Is it true for everybody? So we wanted to see whether a short-term diet intervention could actually help us uncover, someone's microbiome phenotype, response phenotype. So again, I did warn you, we would see a lot of these crossover design figures. So in this case, what we did was we had a diet intervention where we fed people a fast food diet or a Mediterranean diet, and they were randomized to order. They were fed everything in this case. So they were given all of the food in the case of the Mediterranean diet. And on the fast food diet arm, they were given essentially gift cards to specific dollar amounts to buy very specific meals at a Carl's Jr. that was on campus. And you can see that, the diet composition was really, really different. So first of all, the fiber amount in fast food was very, very low, whereas in the Mediterranean diet, it was much, much higher, double of what's recommended, but might be kind of what we all need. And obviously differences in fat amount and percent of saturated fat as well. And again, we control for all of these things every time we do one of these studies. Females all tested during the same phase of the menstrual cycle. All of those inclusion-exclusion criteria, we always apply. No one's allowed to drink alcohol in the 24 hours before they come in for the study visit, et cetera. So just to show you guys what the foods actually looked like, here's a sample meal plan for a fast food diet arm. This is for someone who's eating 2,400 calories. This was all isocaloric, and we made sure that everyone was getting the right amount of calories for their body weight and their utilization. So essentially we fed them, a Pop-Tart in the morning to make them hungry enough to eat 2 giant Carl's Jr. cheeseburgers, 1 for lunch and then 1 for dinner. And then on the Mediterranean arm, they ate a lot of, high fiber foods like high fiber cereal with, nuts and various salads, trek mix, minestrone soup, or whole wheat pasta, extra virgin olive oil. So kind of your typical Mediterranean diet with high fiber and lots of nuts and seeds and things like this. Just as a note, all the study subjects had pretty much no problem eating all of their fast food diet, but complained a little bit about the volume of food that they needed to eat on the Mediterranean arm, which I think is really interesting because, of course, they're isocaloric. So as you can see, the diversity really didn't change, and we sort of didn't expect it to either, because these are healthy individuals who have sort of normal microbiome diversity to start with. So there was high inter-individual variability, though. People were generally more similar to themselves than to others on the same dietary arm. And you can see, some people really did not change at all. Their microbiome stayed pretty much exactly the same, regardless of what arm they were on, whereas other people did see some changes in the microbiome. So there were some aspects that were very predictable, and many others have found similar things. In fact, Peter Turnbaugh's paper from a couple of years before we published ours also found the same thing, that when you give a higher fiber diet, you see an increase in fiber fermenting microbes. This makes perfect sense. And when you give a protein and fat degrading, high protein and high fat diet, you see more protein and fat degrading microbes. So this is all kind of what we expected. But there were some very interesting things, and this is kind of now back to Bilophila wadsworthia in terms of response. So as you can see, this was highly variable. And of course, those individuals who had no Bilophila wadsworthia at baseline didn't see an increase in it. That makes sense. We also saw some people who had low Bilophila wadsworthia at baseline, but who had really large increases after the fast food diet. Some of them were just huge hyper responders. I think if I was subject number 7, I'd be very concerned about eating a high saturated fat diet. And if I had, for example, a family history of various intestinal diseases. And then people with high baseline Bilophila wadsworthia had surprising decreases after the fast food diet. And so we were really intrigued by this, because how could this be? How could you have a decrease when we know that, this microbe is increased by saturated fat? Well, it turns out, that there was a very strong relationship between Bilophila abundance and baseline or background saturated fat intake in the diet. And actually, some of these individuals had background diets that were higher in saturated fat than when we put them on the fast food diet. So essentially, we can detect, who harbors Bilophila wadsworthia and determine whether someone is hyper-responsive to saturated fat content in terms of change in Bilophila wadsworthia. This can be done in a simple 4-day intervention. So this could be something that could be applied clinically, something similar to the idea of an oral glucose tolerance test, where you give a bolus amount of glucose and then you monitor blood glucose levels over the next several hours. In this case, you could sort of give a diet intervention and collect stool samples before and after, and actually see if someone is hyper-responsive to those kinds of changes. So this could be something that we could use to fine-tune saturated fat and fiber composition in the diet to minimize B. wadsworthia abundance, for example, in those individuals who might be particularly susceptible to various IBDs or things like this. So the last thing I want to talk to you about is the effects of not eating. So we actually became intrigued by prolonged fasting because I had a student who came in and was very interested in the anti-aging effects of fasting. And so we designed a not quite randomized order study. This was a, set sequence, but it also had, this kind of 4 time point layout. So basically what we did was we recruited 20 male and female healthy young participants. Again, females at the same phase of the menstrual cycle, diet, meds, physical activity, everything was stable. Plasma glucose was monitored throughout the study, and we had 100% compliance. And what we did was we had people come in basically after a standard overnight fast. So this would be the baseline time point. And so this is 10 to 12 overnight fast. Then they ate their normal foods. Again, we never want to intervene with a standardized diet because that definitely changes people's microbiome, and then you'd need to stabilize them on that first. So instead, we just had them eat their normal foods, but we had them track every single thing that they ate or drank during that day. And then they ate their evening meal, so their dinner at like 6 p.m. They came in for a postprandial blood draw at 8 p.m. They started fasting at this point, fasted the whole next day and overnight, and then came in at time point C at 8 a.m. on day 3 at the 36-hour fasted state, and then ate exactly the same things that they had recorded eating on that first day on day number 3, again had dinner, and then again came in for a postprandial blood draw at that 2-hour postprandial time point. So what we found was that there was a robust effect on the plasma metabolome. You can see that the fasted time point here is clearly completely separated from the other 3 time points. We saw very highly significant changes in many, many metabolites, many of the sort of expected things, for example, increases in free fatty acids. What we also saw, as expected, was increases in all of the ketone bodies to levels that would be expected in someone who is in a 36-hour fast. But we also observed some really interesting changes in specific metabolites that turns out were found to synergistically decrease macrophage activation and inflammation. And then we pursued some additional studies with these metabolites that I won't go into today because they don't really pertain to the microbiome. But what we also saw was that this 36 hours of fasting decreased plasma concentrations of some beneficial microbial metabolites. So unfortunately, you can see here, IPA basically went down to practically 0. And also things like amino acids like tryptophan, which is a precursor for the production of many microbial metabolites in the gut, also really decreased significantly at this 36-hour fasted state. So we were sort of intrigued by the question of whether prolonged fasting could be bad for the gut, maybe by changing the microbiome, suppressing microbial activity, gut barrier function. And then we were curious, are there some people who are more susceptible to the deleterious effects of prolonged fasting in these gut microbiome parameters than others? And so I'll just be able to show you like one result and sort of a teaser here, because unfortunately we did have stool samples from this study, but basically messed up and don't have data from them. They were very difficult to collect because, and we sort of expected this, but didn't realize that it would be so universal. 36 hours of fasting really suppressed bowel movements, and people just weren't really able to produce too many stool samples throughout this time period. And so we really just didn't have enough for sequencing and weren't able to do it this time. So we plan to do this study again, kind of bigger and better, and try to collect those next time. But what I will show you is LPS binding protein here is essentially a measure of gut permeability. So this figure is from a paper where they looked at the amount of LPS binding protein, or LBP, in individuals with SARS-CoV-2 or COVID infection. So LBP is increased in many disease conditions associated with inflammation and poor gut barrier function. So everything from acute infection to sepsis to CVD, obesity, et cetera. Here, just showing you how LBP goes up in individuals with severe COVID. It's universally used as a surrogate measure for essentially LPS in the bloodstream, because LPS is very tricky to measure. So here, showing you just separating male and female subjects and looking at plasma LBP, you can see that at baseline and in the fed state, the female subjects had higher plasma LBP in both states than males in our cohort. But there was essentially no change in terms of from a simple overnight fast to the fed state. Really, LBP did not change much in these individuals. However, what we were really surprised by was that at this 36-hour fasted time point, females had more than a double, 2x increase in LBP at the 36-hour fasted time point no change in the male participants. And then in males, you see a big increase in LBP at the refed time point, but no change in the females. So prolonged fasting has lots of beneficial effects on metabolism, on immune function, but there may be some temporary deleterious effects on the gut microbiome and on gut barrier function. So we definitely found a reduced production of sort of these beneficial microbial metabolites. We did see some increased permeability to microbial toxins, and we saw that these effects might be really sex specific. We were very struck by the fact that the females had higher gut permeability during the 36-hour fast, whereas the males had higher gut permeability during the refeeding period following the 36-hour fast. So this is really kind of highlighting the importance of dynamics in gut microbiome studies. So, I hope I've shown you that careful study design and participant selection is critical for discovering relationships between diet and the gut microbiome. That you can detect even, subtle changes in diet and their effects on the microbiome by, using these tools that we have to really reduce noise. And that short-term dietary challenges, even as short as 4 days, can actually help us to discover individuals who are susceptible to blooms of pathogenic microbes in response to specific dietary components. And then again, the sex-specific effects and dynamics, I think, are really important to focus on too. So just one quick teaser here to put in at the end, because actually most of our funding now comes from grants to look at HDL particles. Why I got kind of sidetracked in this area is because it turns out HDL particles carry small RNA. So Casey Vickers' work from several years ago showed not only do they carry small RNA, but actually most of the small RNA on HDL particles are of microbial origin. And actually a little known or appreciated fact, 30% of plasma HDL are actually coming from the intestinal tract, the upper intestinal tract. And, HDL particles are kind of known for their involvement with cardiovascular disease, but they interact with all cell types throughout the body and probably even cross the blood-brain barrier. So HDL could be this really interesting potential carrier of gut signals or gut microbiome signals and information. And whether this is a good or bad thing is a really interesting question because, although we know HDL particles are very highly protective against a lot of diseases, there also has been this interesting discovery of a U-shaped relationship between HDL cholesterol and mortality. So it's not necessarily the case that more HDL is necessarily better. So I think that this question mark of what HDL might be carrying from the gut is a really important one. So just to acknowledge my lab members and former lab members and some of our funding sources, as I mentioned, most of these new funding sources here are on HDL, and these are the funding sources here who supported the work that I showed today. And I'd be happy to answer any questions.
We do have one question online. How did you track diet adherence to make sure that what you thought they were or were not eating was actually accurate?
Well, in the case of the fast food study, we actually had them bring in all of the containers. So we gave them all of the containers of food, and then we had them bring everything back. And we weighed everything, that was remaining and essentially asked them to eat everything in the containers. For some of the other studies, we basically just essentially asked them to maintain their normal background diets and kept track of their background diets through 3-day diet records and made sure that there were no changes. And then in terms of whether it was the prebiotic supplement or, the eggs, the prebiotic supplement, we gave them the packets and then again had them bring back in all of the empty Ziploc bags and fill out questionnaires. And then for the egg study, again, we actually made and gave them the egg breakfasts and had them bring back the empty containers.
Thank you so much. Any questions in the room? Peter.
Yeah, thanks, Angela. That was really interesting. I was curious what you think about like controlling for background or habitual diet when you enroll the subjects. Like, do you think we should be stricter in like narrowing the range of starting diets?
We definitely do that. Like we ask, do you follow any, specific diet regimens like ketogenic diet or vegan diet or, whatever it might be. We definitely ask about that, and we exclude individuals who have these different diets because, as you see, our approach is really to essentially recruit individuals who are as similar to each other as possible to then try to really, reduce the noise variables and see the effect of the intervention, rather than to recruit large numbers of individuals and then try to account for some of these differences statistically. But that would certainly be the other approach to do that. If you have enough people who are on the ketogenic diet, then you can group them or just make sure you have enough of them so that you can account for that as a variable.
Yeah, I guess I was thinking more like less in terms of special diets like ketogenic, as opposed to like just the variation in macronutrient intake. You mentioned that in the fast food study, the starting level of saturated fat was really variable. Would it have been better to only recruit people that are low?
Yeah, I think that depends on the question that you're asking. I'm glad we didn't exclude people who had high saturated fat intakes because we probably wouldn't have seen these interesting differences in bilophila response. But if, the aim is to see whether individuals who are on low fat diets, when they're put on a high fat diet, have an increase in bilophila, then you definitely would want to recruit people who are on low saturated fat diets. So we always, restrict or design our inclusion and exclusion criteria to be really, really specific to the question. For example, for the prebiotic study, it was like, okay, we really want people who are eating low fiber diets, and we weren't as concerned with the other macronutrients because the main question was the fiber intake question. But yes, these studies are very, very, very tricky. We have started to ask questions even like, do you, taste preferences? Because when we were doing the fast food study and we designed the salads, some people would be like, oh, I don't want garbanzo beans. I want kidney beans instead. And it's like, well, you can't have them because, although from a micronutrient or macronutrient composition, it wouldn't really alter things. It certainly does matter to the microbes, whether it's garbanzo beans or kidney beans, right? So yeah, it's very, very tricky especially when you're giving foods. When you let people eat their own thing, then it's not as big of a problem. But when you're going to feed them something, you really have to ask them, give them a list of foods and say, will you eat this?
Okay, thank you. I actually have one final question. In terms of health, were you looking at oral health as well, or is it just neck down?
Oh, we've just been focusing on gut microbiome, but I think the oral microbiome is very interesting.
So, sorry, I meant like in terms of like recruitment of, you said like young, healthy individuals, like was there oral health factored in terms of what we're considering health, or is it like general systemic health?
Yeah, no, we weren't smart enough to think of that. But it's probably a very good idea, right? Because if someone has gingivitis, that's not necessarily a thing you want to not know about. Yeah.
All right, thank you so much.
Mm-hmm.
So we're going to take a little break just until 3:50, so you have about 10 minutes.
Hi.
Michael. Nice to meet you. Just, how do you pronounce your name? Hatno. Okay. A couple of permutations that I was thinking about.
Yeah.
Okay, great. So I'll just briefly introduce you. Okay.
And you've probably seen in the front, I'm in the front with just a 5-minute, 5-minute warning.
Thank you.
In case you need it.
All right, cool.
Great. So welcome back to the last session of the day. My name is Sergio Baranzini, and I'm a professor in the Department of Neurology here at UCSF. My research is in the microbiome, and half of the lab is computational. So this has been a real treat so far. So we'll continue going strong. And now my pleasure is to introduce our next speaker, which is Michael Patnaud. Michael is an assistant professor in the Department of Microbiology and Environmental Toxicology at UC Santa Cruz. He received his PhD here at the UCSF. His lab now studies microbes that live in the human intestine, and he's interested in metabolism, immune system, and susceptibility to infections. The lab is currently trying to understand the basis of nutritional competition between beneficial and pathogenic gut bacteria for dietary nutrients, and how immune cells influence the outcomes of these competitions. So, Michael, please.
Well, thank you so much for that introduction, and thanks to Susan and Peter and the other organizers for bringing us all together. This has really been an amazing set of talks, and really happy to meet some old friends and new people as well. So as Sergio mentioned, we started our lab in 2020 at the UC Santa Cruz, and we're interested in how microbes compete or cooperate to break down components of human diets. So this image, I like to start with this. This is a cross-section of the mouse cecum in plasticized and then imaged with fluorescence microscopy. The colorful circles that you see embedded in the luminal contents here are beads that we've coated with defined collections of dietary glycans and then gavaged into the mice. So we initially used these beads to measure microbial metabolism. Apologies for brief technical difficulties. It's full, yeah. Hopefully it's not going to sleep on me. Let me just make sure I don't have a screensaver set. Okay, hopefully this will help. Thank you. Okay, apologies. So the beads are coated with sugar. We put them in the mice, and we've been using them to measure how the bacteria degrade that sugar. But since sort of designing this tool, oh, thank you. Since designing these, what we call artificial food particles, we've began to use them to understand how microbes are organized in space. So there are landmarks in this environment, and we're curious how microbes might arrange themselves around these types of nutrients. And we want to know whether that impacts how microbes interact with each other as well. So the benefit of going after all these wonderful talks today is that a lot of the background has been covered. So you're all very familiar with these ideas. The proteins, fats, simple sugars and starches in our diets are pretty efficiently absorbed in the upper GI. And it's this plant material, these polysaccharides from plant cell walls that end up concentrated in the distal gut. And this is dietary fiber, as we just heard about from Angela. The structures of the carbohydrates that make up dietary fiber are diverse. They have diverse monosaccharide compositions and linkages between monosaccharides. And that diversity is important because even a simple change, stereochemical change shown here, necessitates totally different enzymes to break apart the glycans. And when you look at human genomes, we just don't have the hydrolases in our genomes to break apart the full complement of diverse glycans that exist in plants that we eat. Gut microbes do have these enzymes, and they've evolved to use these glycans as a nutrient source. Some species devote large portions, up to 10%, of their genome to the breakdown of plant polysaccharides. So this is exciting. It presents an opportunity to use particular plant glycans to maybe promote species of interest, maybe for species that have beneficial effects on the host. But it turns out to be pretty difficult in most cases to predict which species are going to respond when we provide a given type of polysaccharide. And this is because, as you probably would predict, given the diversity of microbes in the human gut, there's redundancy in the ability to break down polysaccharides. So we had some clear examples of this redundancy in work I did as a postdoc in Jeffrey Gordon's lab. We were feeding mice a whole bunch of different sources of dietary fiber. And although we could stimulate individual species in a model community with particular sources of fiber, when we removed that dominant responder, other species would swoop in and expand in its place. So that's functional redundancy. And it's an issue if you're trying to target your organism that you want to target. So since starting my own lab, I've been really interested in what are the other layers of competition that are involved when two species can both consume a particular plant polysaccharide. So we're curious how the immune system affects these types of competitions, but today I just wanted to talk about some work in progress that focuses on a different idea. The idea is that microbes in the gut might be able to control their localization around these nutrient sources, and that microbes that can increase their proximity to a source of nutrients could benefit. So we're inspired to pursue this question by some amazing work, which many of you have probably seen in the oral microbiota. So Gary Borsi's lab and Jessica Mark Welch in particular have done these fantastic classy fish experiments showing that in human dental plaque, there's this organization. The microbes are positioned non-stochastically, and their arrangements are dictated by adhesion. And so we wondered whether adhesion might provide the type of tools for microbes to control their localization in the gut as well. So plant cell wall fragments, which we've been talking about, are a coveted nutrient source for gut microbes. They're enormous compared to microbes. They could serve as a site where multiple species and many different microbial cells could all come together. And they are distributed randomly in the gut based on mixing. So there's a lot of stochasticity there. However, a lot of gut microbes are non-modal. So how would a microbe increase its proximity to this type of nutrient depot, you could call it? Well, adhesion is one way. So there's some precedent for this. Cellulose degrading microbes in the rumen adhere to crystalline cellulose. That's a micrograph of this process on the right. And that's important for their degradation of plant material. But cellulose degradation isn't thought to be that common in humans. There's also other examples. So bifidobacteria, which we just heard about, actually physically adheres to starch granules, and bifidobacteria are effective degrader of starch as well. And then Bacteroides, which are really abundant in healthy adult humans, can bind non-specifically to plastic and glass. So these are biofilm studies where Bacteroides actually adhere and form biofilms using a particular type of extracellular pilus. And this was pretty recent work by Jean-Marc Guigo. But what we wanted to know was whether abundant microbes like Bacteroides could adhere specifically to the carbohydrate structures that they're adapted to consume. Is there any glycan-specific adhesion observable? We didn't know where we would find such adhesion or how common it would be. So we had to design kind of a screening approach to ask this question. So we took populations of microscopic paramagnetic glass beads, and we coated them with preparations of dietary glycans. So some of these glycan mixtures were very pure. Some were really crude fractions of dietary fiber from a given source, like wheat bran polysaccharides, for example. So each pool of beads has a different mixture of glycans coated on its surface. And then we labeled each of those bead types with a unique combination of fluorophores. Mixing them all together creates a single pool where every color tells you which carbohydrates are present on the surfaces of the beads. And then we could incubate that mixture of beads with individual bacterial strains, in this case in 96-well format. The strains we chose were 160 isolates from human patients belonging to Bacteroides and also Parabacteroides. When we measure each of the beads with the flow cytometer, we can identify which carbohydrate is on the surface. And then if there is additional fluorescence in this pink channel, that indicates that bacteria are bound. And that we get a quantitative readout of binding. So what we found was that binding to specific carbohydrate surfaces among this collection of human gut Bacteroides was widespread. So there were about 20 strains that we tested that aren't shown here that bound to everything. So these are just non-specifically adhering. They bound beads with no carbohydrate coating, basically. So those might be biofilm formers. There was a lot of B. fragilis and B. vulgatus, now P. vulgatus. But this collection of 60 strains appeared to have more selective binding properties. So if you look across the rows, those are the strains. Along the columns, and you can check the bottom, those are the different carbohydrates that were tested. They come from vegetables, fruits, grains, and there's also some host carbohydrates in there as well. So I just pulled out a few just so that you don't have to stare at that dizzying heat map. You can see that these strains have pretty different binding phenotypes. And I should have mentioned binding is indicated by a maroon to black color in the heat map. So, I should also mention we can verify the flow cytometry signal by doing microscopy, that's shown on the right. So, a talented graduate student in my lab, Bo Wei Chang, decided to tackle two questions related to this initial observation. He wanted to know, does this binding actually benefit the microbes? Does it help them to acquire nutrients from these surfaces? And then secondly, because he had seen strains, like multiple strains that have the same binding phenotype, are those strains more likely to interact with each other? Because they're neighbors, if they're reproducibly stuck to the same surfaces, would they be more likely to have an interaction? So to tackle the first question, Bo's approach was to try and identify the genes involved in adhesion. Because if you can mutate the genes, then you can ask, what's the consequence for the fitness of the bacterium? So there were a couple different cell surface structures that could be involved in this adhesion. The first one is these polysaccharide utilization systems that were originally described by Abigail Salyers. These include outer membrane carbohydrate binding proteins. Typically, they're thought to bind soluble polysaccharides, but they could theoretically mediate the adhesion of microbes to carbohydrate surfaces as well. The second structure that we need to keep in mind is the capsule. So these are polysaccharides that are actually made, they're synthesized by the bacteria and produced on the surface. And you can probably see by this EM, which is taken by Eric Martens, who's done a lot of work on these capsules, you can see that the access of the bacterial protein that binds carbohydrates to the surface that it's adhering to, it needs to take into account whether there's this capsule there. And then lastly, the pili that I mentioned previously that have been described, again, relatively recently in Bacteroides, that was our favorite candidate as a potential adhesion structure. These pili have tip adhesins. They're similar to type 1 pili in that they have a tip adhesin, and those usually have a lectin domain, but it's not clear what the ligands for those lectin domains are. So Bo decided to check out these systems, and I'll tell you now, this is work in progress, so he doesn't have the answer yet, but I'll tell you what we've seen so far. So he started with the notion that these polysaccharide utilization systems, they are induced, they're upregulated when bacteria are exposed to the glycans that they are involved in processing. So he did a simple nutrient screen. He took one strain of B theta, which adheres to galactan beads, but not other types of beads. In this case, he's only testing 4 different types of beads. Adhesion is on the y-axis. And then the conditions he tested include growth with a number of monosaccharides as a sole carbon source. He also included maltose, a disaccharide. And that was fortunate because inclusion of maltose actually induced the bacterium to adhere really strongly to this galactan-coated bead particle. So we thought this was pretty interesting. Maltose is known to induce the Sus system, the canonical starch degradation system in B theta. But the type strain of B theta, which puts up a lot of Sus E and F on its surface when you grow it in maltose, it didn't stick at all. And so that really kind of argues to us that it's not likely to be the canonical Sus system that's mediating adhesion. But Bo needs to check that formally. Just as an aside, we were initially confused about why maltose, this disaccharide, would induce adhesion to galactose, to galactan, which is polymer of galactose. But if you look at the structures of the polysaccharides on the right, although the monosaccharides are different, the glycosidic bond is really kind of just flipped. And so the monosaccharides may adopt a really similar conformation in these 2 structures. Yeah. Okay, so we don't think it's these stubby carbohydrate binding proteins in the Sus system that are involved in the adhesion. So what about the capsule? So capsules are known to be induced by nutrient conditions as well. And so we were fortunate enough to get to meet Eric Martens, and he was generous in giving us a series of capsule mutants where the bacterium expresses only one of its 8 capsules at a time. So this is important because the wild type B theta can switch its capsules through phase variation, and it makes it pretty difficult to study. But Eric has these mutants. And what we found when we tested them was that capsule cells expressing only capsule 7 or only capsule 8 were strongly adhesive. They bound to galactan with high affinity. And there's galactan down there at the bottom. There's also a second source of galactan with a small pactic fragment on it. And they both showed similar binding profile. And then here, Bo's using a larger library of carbohydrate beads. So you can really appreciate how specific the adhesion is of the cells to these galactan polymers. Okay, so this really suggests that the capsule is intimately involved in the adhesion phenotype that we're studying. And we are actually tempted by the idea that the capsule itself mediates some sort of carbohydrate interaction. It seems far-fetched given the specificity. And in addition, there's a couple other pieces of evidence that argue against the capsule actually mediating adhesion. Those include the fact that maltose also induces increased adhesion in cells that have locked on the capsule, and we don't detect any increase in the amount of capsule, at least like the transcript level expression of capsule synthesis genes. So the bottom line is, although capsule is intimately involved, we actually think there could be a second factor that's important for adhesion as well. So Bo's taking a few approaches to try and assess what other factors might be involved. So I'll just tell you about those briefly. So he's starting with this, this induction phenotype. He's doing RNA-seq to track which genes are induced by maltose. In this volcano plot, you can see that he's got the starch degradation system clearly induced, as we would expect. Interestingly, there's some genes on the this plasmid in B theta that are also induced by exposure to maltose. But then there are some genes down there in the bottom in red dots that we think could be involved in adhesion. And now Bo is going through one by one and overexpressing these genes in B theta to see if they're sufficient to induce the adhesion phenotype. He's also taking a broader approach. We have the benefit, thanks to Andy Goodman, of doing transposon mutagenesis in B theta. So Bo's got mutant libraries, and you can imagine the type of adhesion-based separations that you could do, separating binding mutants from non-binding mutants, and then sequencing to see what genes are disrupted. So we're hoping to find genes that also repress adhesion, so hyper-adhesive mutants, in addition to genes that are necessary for adhesion. And then where he wants to go in the future is once mutants can be identified that have variable adhesion phenotypes, he wants to put them in mice and see if that adhesion is operative in vivo and whether it impacts bacterial fitness. So one of the main reasons we're using this bead assay to do adhesion is because you can put the beads into mice like we did back in this paper in 2021, and then colonize those mice with a selection of strains that you're interested in and assess whether the bacteria bind to one bead type or another in that context. Okay, so I also wanted to tell you about some more work in progress. Bo is tackling this second question. Do species that co-adhere to the same surface, do they have strong interactions with each other? Have they maybe even co-evolved if their co-adhesion is really reproducible in the gut, if it's always present? So he started to look at this from the lens of metabolism. So what he observed was that a few of the strains that stick to galactan with what appears to be high affinity, they weren't capable of breaking down galactan as a nutrient source. So you can see along the bottom here, he's got a list of strains. These are all strains that stick really strongly to galactan-coated beads. And then in the wax, he's measuring growth. And in glucose in blue, they all grow. And then there are 4 that fail to process galactan when it's provided as the sole carbon source. That's also true over long time scales. So he cultures them for a couple of days, and he still sees that, for example, this strain, see if I can get a pointer working. This strain, 4087, even after 120 hours, has a profound defect in the metabolism of galactan compared to another strain, 3164, that can grow on galactan. They both bind galactan. They stick to it. So Bo was able to sequence the genome of this strain with short and long read sequencing, and he found that the strain lacks one of the critical enzymes in the galactan processing pole in B theta. It doesn't have this GH53. And so we think that probably explains why it can't grow on galactan. It also lacks a GH2, but it has other GH2s in the genome. So we actually think it can probably process oligosaccharides if it gets its hands on them. It grows on galactose, the monosaccharide. And if you provide galactan, the polysaccharide, and then provide an exogenous GH53 in the media that breaks down galactan, you can rescue growth. So it's expected, but it raises the notion that if this deficient strain cozies up to a strain that's making a lot of GH53 or galactose breakdown products, there could be a rescue, there could be a cross-feeding involved. So Bo wanted to test this idea by co-culturing microbes together. So this experiment has the deficient strain on the y-axis. You can see it grows in glucose, it fails to grow in galactan. That's the phenotype. And then by providing galactan in combination with co-culture with a number of other strains, you can see that some of them are able to rescue the growth of this deficient strain on the y-axis again. And then Bo took one of those and did a more careful experiment over here on the right, and yes, verified that there's a positive interaction when these two strains are grown together. And then lastly here, he's able to, oh, thank you. He's able to take conditioned media from this putative feeder strain and add it to a putative, what we could call a putative cheater strain. And he sees a modest effect of adding that conditioned media as well. So we think the rescue could be mediated by a secreted factor. The simplest explanation, we think, is that the feeder is a sloppy consumer of galactan and releases oligosaccharides, which are then taken up by the putative cheater. But to prove that, we have a lot of work to do. And Bo is starting with deleting this galactokinase enzyme in the cheater strain to see whether galactose is involved at all in this cross-feeding. It's the only galactan is the only carbon source in the media, so we feel like galactose is probably involved, but he needs to do the experiment. All right, and then in the last slide here, I just wanted to give you an idea where Bo is headed with these projects. So he wants to take a much broader view and determine whether other species, other taxonomic groups in the gut adhere to dietary particles. So he's taking large libraries of these carbohydrate-coated beads and gavaging them into mice. Here you can see that the floors we've chosen survive in the mouse GI tract. The ratios of bead types are the same after we pull them back out of the mice. And by sorting them and doing 16S sequencing, he wants to figure out which species alight upon different surfaces and do they form communities of microbes that might interact. And then lastly, we hope to eventually engineer interactions between microbes that might be weak at baseline or might not even exist. By exploiting these adhesion systems, you can change whether the bacteria express certain adhesion proteins and then change the diet by including or disincluding certain surfaces. And we want to bring together species that might interact. And we have some type 6 secretion system positive strains that could be good candidates for this type of system. And with that, I just want to point out again that Bo Hui is the PhD student who did this work. I want to thank all the members of the lab and especially our collaborators, Eric Martens at University of Michigan, Ashley Wolf at UC Berkeley. And a lot of the initial adhesion screening was done in the lab of Jeffrey Gordon when I was a postdoc. I'm happy to take questions.
Thanks for a great talk. I was curious if you've looked at the protein corona of some of your beads to see if there's different adhesins that come on, and that might be shunting different bacterial adhesions.
We haven't done, I think you're suggesting to use the beads biochemically to isolate proteins that might be bound?
Yeah, and if those proteins are causing certain bacteria species to overbind, as opposed to just the glycan itself.
Oh, yeah, yeah. Yeah, so the attachment chemistry that we use is not glycan specific. So it's possible that there's, especially in the crude fractions that we tested, there might be proteins that, yeah, serve as adhesive ligands. So we've followed up with several of our beads that have really strong binding with a carbohydrate specific attachment chemistry to test whether that were proteins involved. And it seems to be carbohydrate specific in the cases we've tested. But yeah, it's a good idea.
Do you see any interaction between immune cells in the gut with your beads and microbial communities that are on them?
Yeah, that's a cool question. So I think neutrophils are probably, people have studied neutrophil sort of transmigration into the gut lumen. We haven't looked at that at all. I guess that's where we would start with that. Yeah, so we haven't checked yet. Yeah,
okay, then I have a different follow-up.
Yes.
Do you have a prediction of what you might see, especially since it looks like you have heterogeneous sizes of the beads that are going in? Do you have a sense of what you might see and whether or not that's going to influence the competition within bacterial cells themselves?
Yeah, it's an interesting idea. Still considering immune cell binding? Yeah. Yeah, I think maybe the thing that interests us in that realm is whether the beads actually make it across the gut epithelium. So we have started to look at that. These beads are too large, but we can make smaller beads that are around 1 micron or half a micron in size. And yeah, we're really interested in M cell, whether M cells, for example, would transport beads coated with different surfaces. And the screening approach maybe provides a high throughput way to test for that. And then we want to know how would they interact with immune cells. Yeah, thank you.
Yeah, I was curious if you've looked at like how the glycans get chewed off the bead. Like, do they get eaten away from the outside, or can you get like large glycan pieces that slip out?
Yeah, that's a great question too. The metabolic experiments we've done to assess how the bacteria chew down the polysaccharide, they're all loss of signal assays. So we measure the beads after they've been incubated with microbes or put through the mouse. So whether like it's getting chewed down from the top or whether it just gets clipped at the bottom and is gone, we can't distinguish. So we haven't done like a more careful assessment of that. We've thought about it with antibodies that bind to the carbohydrates. Maybe that might be a cool way to do it. Yeah.
All right.
Yep. Thank you.
And our next and last speaker for the day is Benjamin Good. Ben is an assistant professor of applied physics in the Department of Applied Physics at Stanford. He received his PhD in physics from Harvard, and he was broadly interested in understanding the ecological and evolutionary dynamics that control the structure and function of biochemical cycles driven by microbial communities. The Good Lab uses tools from statistical physics, population genetics, and computational biology to study how microscopic growth processes and genome dynamics at the single cell level give rise to the collective behaviors that can be observed at the population level. So, Ben, welcome.
Awesome. Well, thank you for that kind introduction, and thanks so much to Susan and Peter and all the other organizers for inviting me out to give a talk today. It's a real pleasure to tell you about some of our recent work looking at the population genetics of our gut microbiome. It's a little tough going after so many other good talks today, so I'll try to keep it brief. So of course, one of the main challenges in understanding microbial communities like the gut microbiome is that they involve a large number of ecologically interacting strains, often with enormous complexity when we zoom in and look at the microscopic level. Got another great image from Jessica Mark Welch and Gregory Ford here. Of course, another challenge, the one I actually want to talk about today, is that the individual actors in this game aren't necessarily fixed and can actually evolve over time by acquiring heritable mutations in their genomes. And this process can happen extremely rapidly in microbes, as we all know, with some well-documented examples in pathogens and laboratory experiments showing evolutionary changes sweeping through microbial populations on timescales as short as years and months, and in some cases, even days. And if this behavior is sufficiently widespread, you could imagine it would have important implications for efforts to understand or eventually manipulate communities like the gut microbiome, since it means that these ecological interactions could be constantly shifting under our feet. But in contrast to the ecological dynamics that are going on at the species level, which I'm kind of illustrating in different colors here, we currently know relatively little about the evolutionary processes that are going on within individual species in big microbial communities like the gut microbiome, to the extent that even a lot of basic phenomenological questions still aren't really well understood. So for example, what are the relevant timescales for within host evolution? A week, a month, a whole lifetime? do we expect these resident populations to evolve much at all, or are bigger communities just better able to respond to environmental perturbations through purely ecological means, right? So shifting the abundances of the species that are already there once they see some perturbation, rather than waiting around for one of the resident strains to acquire some random mutation. The resident populations do evolve. What do the population genetics of this process look like? What is the genetic structure of a typical resident population in a person's gut? How important are factors like genetic drift, horizontal gene transfer, or immigration of new strains from outside the host? Then finally, how do these short-term processes within hosts relate to the longer-term evolutionary processes that shape genetic variation across hosts in the broader human population? I'd argue that without knowing the answers to these really, really basic questions, it's hard to know what sorts of evolutionary models we should be thinking about in the first place, or how we'd go about rationally designing or interpreting data from experiments to try to probe these questions. And so a lot of our work is motivated by the observation that at this early stage, even some really rough order of magnitude estimates about these different evolutionary forces could be really useful for helping us, motivate future theoretical or experimental. And so to try to address this question, one of the things our group's been doing is developing approaches for going in and measuring the evolutionary dynamics that take place in a human gut microbiome using shotgun metagenomic sequencing of human fecal samples, right? Where we just take the entire collection of bacterial genomes that are present in a fecal sample, chop them all up and sequence them in these big pools of short sequencing reads. So in principle, these data should contain an enormous amount of information about the genetic diversity present in each of these populations, since to a first approximation, each one of these sequencing reads is effectively sampling from a unique cell. But extracting this information is still a major challenge. That's because the genetic structure of these resident populations is generally going to be shaped by a combination of different factors. Right, so we can imagine different bacterial lineages are going to enter the host during initial colonization sometime in childhood and potentially through subsequent migration events later in life. And then once these genomes are in there, they're going to start to get shuffled around according to population genetic processes like mutation, selection, recombination, and genetic drift. Then, of course, these seeding lineages themselves are drawn from some broader global population of gut bacterial strains, which is shaped by its own set of population genetic processes. And this means that the collection of bacterial genomes we observe in a given fecal sample today are going to reflect a mixture of all of these different forces. Right? We just got done saying we don't understand that much about them yet, so it's a little bit of a catch-22. To make matters a little worse, we rarely so privileged to be able to observe these different bacterial genomes directly, but only their sort of image, so to speak, built into these big pools of short Illumina sequencing reads. So what we can measure really well are the frequencies of individual single nucleotide mutations, similar to what Jose talked about in his talk, for example, by aligning these short sequencing reads up against the reference genome or an assembly of a given species. And then looking for locations where you observe a mixture of two different bases at a particular site. So this little cartoon I'm drawing here, we infer there must have been a mutation between T and A at this particular site in the purple species genome. And because the A's are present in 3 out of the 5 reads that line up to this site, we infer that the A mutation must be present at about 60% frequency within this particular species in this particular sample, right? Perhaps with some Poisson measurement points. So our general approach then is to try to use mathematical models from population genetics to help us interpret the noisy statistics of these mutation frequencies directly. And in doing so, see if we can pull out some reasonably quantitative estimates about these different evolutionary forces without first solving this complicated metagenomic inverse problem. Let me just give you a brief flavor of how we go about doing this. So one of the simplest things you can do with this kind of data is just aggregate all of our mutation measurements across the genome of a given species, then ask how much the total amount of genetic diversity varies between different resident populations in different fecal samples. This is actually very similar to what Jose was showing in his talk earlier. So here I'm just showing you the total number of intermediate frequency mutations in the Bacteroides vulgaris populations from 700 different fecal samples. And I've zoomed in, and here I'm showing you the distribution of their frequencies within that species for 3 particular hosts from this panel. So you can see that these data vary over almost 5 orders of magnitude here. Right? I sort of plot them on a log scale on purpose. Some individual fecal samples, the B. vulgaris populations, might only have a few mutations across their genome, while others might have tens of thousands. Right? So using what we know about the typical mutation rates and generation time to take place in the gut, you can do a little back of the envelope calculation to convince yourself that many of these hosts over here on the left have way too many mutations to have accumulated over a single host lifetime. And that means that we can't treat each of these populations as being descended from just a single founder strain. At the same time, In these two examples here, you can see that there's quite a bit of variability between these two hosts. In fact, too much for us to be able to treat them as some large random sample from some broader population, somewhat akin to what we would do with a city in population genetics. Instead, these data tell us that we force us to consider an intermediate scenario, which we sometimes call oligocolonization, in which individual hosts can be colonized by just a handful of external strains that enter and grow to reach appreciable frequencies. Right, and the mutations that accumulate between these two strains before they colonize the host then show up as these little bumps in their mutation frequency distributions that I'm showing you here. This is arguably the worst possible scenario if you actually want to measure genetic differences between these different strains, right? What we want to start out with. And that's because it's going to require us to assign individual mutations to the lineages in our little cartoon here. And this is going to be a hard inverse problem because we're rarely going to catch these mutations together on the same physical sequencing read, at least with Illumina sequencing. But from the examples I've chosen here, I sort of want to convince you that this intractable inference problem also has some really easy special cases. So in hosts like host C over here, you can maybe convince yourself that two mutations that we sample at 99% frequency in the same host are almost surely likely to be present in the same sets of cells, right? So there's no way you can set that up so that it's not the case. And this sort of illustrates our general approach here, which is to try to leverage the considerable variation we observe across a large sample of human fecal samples, and then just zero in only on those particularly easy samples where we can infer the dominant strain with a high degree of confidence. And so with this kind of approach, you're going to have to discard a lot of data in the general case. But fortunately for us, there's been a lot of sequencing done on the human microbiome. And so it's possible to use this quasi-phasing approach to measure really small numbers of genetic differences between thousands of strains in let's say, 10 to 40 of the most prevalent species in the human gut. And this lets us start to bootstrap estimates about these different evolutionary forces. So we've now applied this basic approach in several different contexts, but the story I want to tell you about today is one that's led by one of my graduate students, Jiru Yu. In fact, this is the first time I've talked about this project, so I'd be really curious to get feedback from you guys in the audience. This story actually begins with some previous work that we did in collaboration with Nandita Garud and Katie Pollard and Oscar Halacek, where we examined the diversity of a large cohort of healthy human microbiomes that were previously sequenced by the Human Microbiome Project and a few related studies. And so this cohort we put together consisted of on the order of a few hundred hosts, a subset of whom were sequenced at 2 different time points, roughly 6 months apart. And the idea here was that this kind of design would allow us to both look at short-term evolution within hosts over this 6-month sampling interval, as well as longer-term evolution across hosts in the broader population. I should usually talk about this short-term thing down here, but actually today, I want to focus on these longer-term timescales. Okay. So what do we see? When we take strains from unrelated hosts and compare them, here too, we also observe a really huge range in the genetic differences that we observe between different pairs of strains, both within or between different countries and continents. Some pairs of strains have, again, just one or a few mutations across their entire genome. Other ones have mutations at up to 10% of all their synonymous sites in their protein coding genes. Here I'm just showing you an example for Bacteroides vulgaris. It's one of the most common and abundant species in the industrialized human gut. To first approximation, most of us in this room probably have this species. And you can see that, even though this cohort is relatively modest by 2023 standards, only a few hundred hosts, this quasi-phasing approach already allows us to get a sample size that's bigger than the total number of sequenced B. vulgaris isolates that are out there. And with this larger sample size, we can start to see some interesting trends. So first, at the largest genetic distances, we see that some species, like B. vulgaris, can be naturally divided into two well-defined clades or subspecies. In this case, there's two different major clades, and they're about 10 times more diverged from each other than pairs of strains from the same clade. I think one thing that's kind of nice about this system is that it can be helpful to convert these genetic distance estimates, which can sometimes be a little abstract, into corresponding timescales. And again, using what we know about the typical mutation rates and generation times in the guts, we can estimate that these two clades started to diverge from each other about 140,000 years ago, while pairs of strains from the same clade started to diverge from each other about 20,000 years ago. Just for reference here, these timescales are, let's say, comparable to the out-of-Africa timescales of human history. I think, interestingly, they're quite a bit more ancient than, let's say, modern industrialization timescales, which maybe raises some interesting questions about where all this diversity actually came from in the first place. So if we look at pairs of strains from the same clade, you see that most pairs of strains are pretty distantly related from each other and are consistent with this 20,000 year age we talked about before. This is true even if we look at strains sampled from the same geographical location, like people in this room, and sometimes even from the same host if it happens to be one of the ones that's co-colonized by more than one beagle goddess strain. But the main thing I want to talk about today is the fact that in a large enough sample, we can also find some pairs of strains that are anywhere from 100 to 1,000 to sometimes even 10,000 times more closely related to each other on average, more closely related to each other than this population-wide average. We sometimes call these bacterial cousin strains. But unlike human cousins, I want to point out, we're pretty sure these are not due to some secret relationships between their human hosts. Not only are we specifically looking at unrelated hosts here, but actually you can find examples of these closely related bacterial strains, even many examples, even when you look at hosts taken from different continents. So there's some pretty rapid mixing going on in the human population. Now, given that observation, you might then ask, okay, are these clusters of closely related strains maybe just very recent examples of subspecies, maybe evolving to exploit some more modern dietary? Actually, if we start to examine the genetic differences between these strains, we start to see some weird patterns that maybe aren't consistent with this idea. So in particular, what Dura was able to show is if you look at the synonymous mutations that have accumulated between these two strains and look at where they happen on the genome, they're not randomly scattered across the genome as you might expect, but instead appear in these runs of, let's say, tens to hundreds of consecutive SNPs in localized regions of the genome. In fact, you can show that this kind of behavior is inconsistent with simple models of isolation and divergence through mutations, and instead suggests that a lot of the mutations that are occurring between these strains, the differences that are human between strains, are driven by ongoing bacterial recombination. So what do I mean by that? Probably all familiar with the idea that bacteria can exchange DNA with each other horizontally through several different mechanisms. Maybe I should put vesicles up here now too. Didn't have quite enough space. We usually think of this as a way to acquire almost completely novel genes or pathways, often through transfers from other species. And this can, confer resistance to drugs like antibiotics, or enable novel metabolic capabilities. And there's some, really classic examples of this kind of behavior going on in the gut microbiome in particular. But we also know that DNA can be transferred between more closely related strains as well, potentially at an even higher rate. And in this case, these transfers can actually overwrite existing regions of the genome by a homologous recombination. And this more subtle form of HGT acts to reshuffle genetic variation within species, similar to meiotic recombination in sexual organisms like humans. This allows them to maybe more efficiently explore DNA sequence space. Some classical arguments from bacterial population genetics have shown how this process of homologous recombination can maybe give rise to that block-like pattern of divergence I showed you on the previous slide. And here's a little cartoon of how this might work. So a typical pair of strains shares a common ancestor quite a while ago. And so if there's lots of recombination going on, by now their present-day genomes are going to be a mosaic of overlapping recombination events. Right? So the divergence between them is going to be pretty high and consistent across the genome. If we happen to get really lucky and sample a pair of closely related strains, their genomes are going to be nearly identical to each other, perhaps with some handful of mutations that have accumulated in that short bit of time. So then you can imagine a recombination event into one of these two closely related strains will typically be sampled from one of the more distantly related strains of the population. This is going to show up then as a sort of block of higher divergence against this nearly clonal background. Right? This process can continue with more transfers coming in and more blocks until you get to a point where essentially the entire genome has been overwritten by these homologous recombination events. And then we're back to the mosaic picture that we started out with. And this is sort of our idea for what could potentially give rise to these this block-like divergence pattern we observed in beagle goddess here. And so, just the mere fact that gut bacteria can recombine is maybe not so surprising. Right? People have observed this recombination process in lots of different bacteria. However, the dynamics of this process, I would say, are still really poorly understood in their complex native environment of the gut microbiome. Right? So in nature, even really basic questions about the typical rates or the mechanisms or the evolutionary consequences of these transfer events are still really poorly understood. So given the large number of closely related pairs we found in this data set, we thought we might have the opportunity to start to probe these questions a little more systematically. And so to do this, Giroux developed an automated approach for resolving large numbers of individual recombination events between closely related pairs of strains. And the way he did this is pretty simple in principle. So use the hidden Markov model to classify regions of the genome into clonal regions or recombined regions, just based on their local patterns of sequence divergence, like I showed you on the previous slide. And in some species like B. vulgatus, where we had really well-defined clade structure, you can further partition the recombination events into those that came from the same versus the opposite clade, since they're associated with significantly different levels of divergence on each one. So we then applied this approach to analyze more than 7,000 close pairs of strains from about 30 of the most prevalent species in the industrialized human gut microbiome. Here's just a little example of what we see when we do this. So again, I'm showing you data here starting out for the Bacteroides vulgatus population. It's helpful to actually visualize these data by plotting the total number of detected transfers as a function of the genetic divergence in the clonal regions alone. And the idea is this provides a proxy for time. And so the slope of this relationship then tells us something about the recombination rate that's taking place in these organisms, at least normalized by their typical mutation. So of course, in B. vulgatus, we can further divide these measurements into those coming from the same clade versus those coming from the opposite clade. Just a plot down here, kind of inverted. So this is data from just one pair in our cohort. These are data from all the different pairs. This blue line is showing sort of a running local average. So with this larger number of data points, I'd argue we can start to pick out some interesting trends. So for example, we observe a trend towards a slightly larger number of successful recombination events in strains with larger amounts of clonal divergence, consistent with the accumulation of more recombination events over time. We also find that these recombination events accumulate relatively uniformly across the entire core genome of B. vulgatus. They're not just localized to a few specific genes that are driving the entire signal. So both of these observations are sort of consistent with a qualitative picture of accumulating, steady accumulation of recombination events over time. But the quantitative bits of this data here tell us that it doesn't appear to be accumulating just via some simple linear trend like we might expect. Instead, there's actually quite a bit of heterogeneity between different pairs of strains. So some pairs have anomalously large number of successful recombination events, even at relatively low values of clonal divergence. Other pairs have the opposite trend, so relatively few recombination events, even when they're relatively more distantly related from each other. You can show actually the spread is quite a bit larger than we expect just by a random Poisson sample. We can also see that the rate of recombination coming from the opposite clade is quite a bit lower than recombination transfers coming from the same clade. Suggestion there's some degree of maybe genetic isolation between these two groups, maybe similar to what you'd expect for different species. Now, in principle, this genetic isolation can be caused by several different factors folks have thought about. There could just be fewer opportunities for these two groups to recombine with each other, either because they prefer separate ecological conditions and just rarely bump into each other, right? Maybe they live on different glycan depots. Or maybe by now the two clades are just diverged from each other enough that there's less homology there to initiate recombination events in the first place. It could also be due to more elaborate factors, right? Like incompatibilities on the recombined fragments, or potentially incompatible restriction modification systems, which would chop up DNA from coming from the wrong clade or the other. So we can actually start to distinguish between these different hypotheses by also examining the typical lengths of the recombined fragments, which Giroux can also measure with his computation algorithm. In this case, we see something really interesting, which is that we observe systematically different lengths of the successful transferred fragments, depending on where they came from the same versus the different clade. Right? And this simple observation allows us to rule out really simple models of ecological isolation or limited opportunities for homologous recombination, which we would expect to just scale the overall rates uniformly without changing the lengths. Here's just one example for Bacteroides vulgatus. Of course, we can apply the same kind of approach to all the other species in our larger data set. And this allows us to obtain quantitative estimates of the typical transfer rates and the typical lengths of the transferred fragments in about 30 of the most prevalent species in the human gut. And so if we zoom out, we can use these data to start to see some interesting trends at, let's say, higher levels of taxonomic organization. So for example, many species in the Elistopes genus tend to have slightly higher rates of transfer, involving slightly shorter fragments, where species in the Bacteroides, Bacteroideaceae family now, I guess, because it's been split, tend to have somewhat lower rates of transfer and slightly longer fragments. But we can also observe significant differences within individual families or genera as well. So just as an example here, B. cacchi over here on the left accumulates successful recombination events at a much lower rate than B. massiliensis over here in the middle, which accumulates them much more steadily over time. We zoom in even further, we can still see even more differences within, more heterogeneity, even within individual species. So for example, some species like B. cacchi over here on the left exhibit sort of bimodal distributions of recombination events, where a few strains, have a larger number of successful transfers, but the vast majority of them have maybe 0 or 1. And other examples like, let's see Putridinus over here, you see some evidence for sort of systematically different rates of recombination in different time intervals. So the slope sort of changes from maybe a higher value early on here to maybe a lower value as evolutionary time progresses. And again, here we also observe a significant spread in the amount of recombination, even for strains with the same amount of clonal divergence today. And we can again show this is sort of larger than you'd expect just by chance. And so together, these different observations sort of point to maybe an interesting picture where the dynamics of recombination within a given species of gut bacteria can't be captured by a single recombination rate, which a lot of our evolutionary models assume. I think this raises some interesting questions about what forces give rise to these different kinds of heterogeneity. Okay, so finally, we also wanted to ask whether we could see other examples of this genetic isolation between clades that we observed in the Vivogatus populations from before. And in this case, it requires to know about the clades ahead of time. And so to try to generalize this idea, we instead just tried to quantify the raw genetic divergence values for all the detected transfers. This tells us something about the sequence divergence of the donor fragment. And then we threw all these together into a big bucket and then asked how this observed distribution of donor sequence divergence differs from the expected version that we get, which is randomly simulated transfer events between different pairs of strains. And so the reduction we observe at higher levels of sequence divergence here are just another way of saying that we observe fewer successful transfers between clades compared to within clades. So we zoom out and apply this approach to other species in our data set. We can find a handful of examples like Vivogatus that have a reduction in recombination rates at higher divergence. And actually, in most of these cases, the clade structure was similarly obvious ahead of time. But interestingly, in the vast majority of cases, we didn't observe any evidence for reduction in the rate of recombination with higher sequence divergence. And so this is maybe suggesting that recombination barriers are playing a limited role in most species of gut bacteria. And the fact that we can observe these pretty striking differences, even for relatively closely related sets of species, which live in the same environment and presumably share the same, molecular recombination machinery, is telling us that maybe the exceptions to this rule, like B. feingoldii or B. vulgatus, maybe have an evolutionary rather than a simple biophysical origin. Okay, so just to summarize what I want you to, the main things I want you to take away from this talk. We've talked a lot about heterogeneity within or between species, but if we zoom out a little bit, you can think of this data as giving us an estimate of the typical recombination rate in different species of gut bacteria. It's accurate to within an order of magnitude or two, right? Same thing with typical lengths of the recombined fragments. We can then combine these estimates with previous estimates for the rates of mutation accumulation over time to convert these recombination rate estimates into human relevant timescales again. Right, so if we pick a single bacterial lineage and follow its lineage backwards in time, we expect the first recombination event to occur, let's say on the timescale of about 10 years, right? Could be as short as 1 year, could maybe be as long as 100, but something on that scale. So from this perspective, maybe recombination seems relatively slow. But it's not necessarily slow on longer evolutionary timescales. So if we trace this lineage back further in time, after about 3,000 years, we actually expect the entire genome will have been overwritten by recombination events. And this is about 10 times shorter than the typical ages of circulating strains that we talked about before. This means that in between these two timescales, we expect that these bacterial genomes are going to look quite a bit more like human genomes, and that they're going to consist of a mosaic of overlapping recombination events. So there's one thing I want you to take away from the talk, it's this. It's that on these intermediate evolutionary timescales, gut bacteria look more like mosaics than like classic phylogenetic trees. This is really important for questions like trying to understand how bacteria have co-evolved with different human host populations. This is typically done by constructing a phylogenetic tree for different bacterial isolates and asking how it associates with different host populations of origin. If enough recombination is going on, the problem is that there really is no true genome-wide tree on these intermediate evolutionary timescales. Instead, sort of a collection of different trees that vary locally on the genome. Maybe another way of saying this is, if a strain transmitted from one continent to another more than 3,000 years ago, by now we actually expect its genetic material should be entirely intermixed with its new population and vice versa. And this suggests that maybe we're going to require some new methods, maybe borrowed from human genetics, to try to understand these transmission processes on longer timescales. Okay, then finally, we can also ask whether we can extrapolate this down all the way to the scale of a single host. You might expect that given this estimate here, maybe recombination is a relatively weak force on a within host timescale. But to really do this extrapolation, we also have to account for the fact that each of us is home to a whole population of bacterial cells of the same species, and they're all potentially exchanging DNA with each other at the same time. So once we multiply our estimates by this larger number of cells per person, you estimate that even though the rates are pretty low overall, we still expect that a single person's gut microbiome, single species, produces anywhere from 1 million to 100 million new recombinant cells every day. Or to phrase it a slightly different way, every site in the genome of one of these species is involved in anywhere from 10,000 to 100 million recombination events every day. So even if just a small number of these provide a fitness benefit to the new cell, we expect its descendants should rapidly sweep through the population and fix. And so this suggests that maybe recombination is still going to be important for driving within host adaptation, even if it's too slow to completely reshuffle bacterial genomes in a single person. Indeed, we find some evidence that this is going on. Okay, and then finally, I just want to end by saying that these quantitative estimates actually raise a lot of really interesting open questions from my perspective. Maybe the most important one is, where, when, and how is all this recombination actually occurring? Now, I mentioned we don't really know which mechanisms are at play in real people's guts, but one thing we do know is that most species of human gut bacteria I showed you here are not naturally competent. They don't just take up DNA from the environment. And yet they still recombine almost as much as other species that do. And so this suggests that maybe other mechanisms, conjugation, phage, maybe vesicles, are important for driving these sorts of effects. It's maybe a bit surprising when you think that it'd be pretty costly to sit around and wait for an hour to conjugate with somebody in an environment like the gut, where you really need to reproduce in order to evade evolution. We also see relatively little correlation between the amount of recombination and the apparent degree of co-colonization, which you might think is necessary for these recombination events to occur in the first place. This suggests that maybe intermediate vectors are driving some of the patterns that we're seeing. And then finally, given all of this sort of complicated ecological factors that we think are maybe behind this, it's also maybe a bit surprising why these realized recombination rates that we can detect in strains we sample from people are actually so similar across species, right? You might think that an order of magnitude variation is pretty big, but from another perspective, it might seem pretty small, given that we expect maybe some of these other factors to vary over even larger regions. And this raises some interesting questions about whether maybe evolution itself has sort of fine-tuned these recombination rates to make them lie within this relatively narrow region pinned close to the mutation rate. This is something we're currently spending a lot of time thinking about as well. Okay, so with that, I guess I'd just like to thank my wonderful collaborators here, particularly Narita Garud, who's my co-author on this HMP study I talked about at the very beginning, and of course, Juru, who really led this recombination analysis I talked about in the bulk of the talk. And thank you all for listening. Be happy to take any questions.
Thank you, and it's great to see these numbers actually, from natural samples. When you compare these numbers and you see different numbers between families and between species, were you already able to look at the gene content of these genomes and maybe correlate these differences with difference in terms of the mobile genetic elements present, like specific prophages, specific plasmids, or other kind of mechanisms that could explain why you see different rates, basically, of recombination across this taxa?
Yeah, we haven't looked at that systematically yet, and I guess the reason why is maybe comes back to this plot I showed maybe towards the beginning of the talk. Most of these recombination events are occurring relatively uniformly across the genome. So if mobile elements are involved, they're sort of maybe involved kind of in an intermediate stage, right? It has to get on the mobile element, get into the new cell, and then get back on. This is true even for sort of boring housekeeping genes in addition to the exciting ones. So yeah, we haven't looked at that a bunch. I think maybe now with bigger isolate collections, that would be an interesting angle to go. And the thing we're really interested in, I guess, now is really trying to say, like, this is maybe a baseline for the amount of recombination going on. Can you now see that mobile elements are hopping around faster relative to a given species? Yeah, that'd be awesome. Yeah.
Lovely work. Have you looked in your original allele frequency data to see how sampling depth in those metagenomes affects those by maybe resampling that metagenomic data and seeing if you end up consistently with the same estimates?
Yeah, we haven't looked at that as much for this particular question. We've looked at it a lot for detecting, sweeps of individual mutations within hosts over time. It's maybe worth mentioning that essentially what we're trying to do here is only look at the cases where we're like super confident we can say what's going on. So we purposely only look at the highest coverage ones, and then even there, only the ones where it's just one strain at a dominant frequency. So all the ones that are sort of mixtures of strains where coverage really matters, there we have to go back and be really careful sort of think through it.
That's very similar to what we've had to do.
Yeah, I think that's maybe the only way to really, without long reads.
There's some other methods that are coming out that may be a little bit better at predicting, but the other question I had is within the differences that you see in some of the species, do you think that has something to do maybe with the fact that it's not a monolithic ecosystem within the gut, that maybe some of these organisms are actually splitting niches within the gut, in the lumen versus close to the mucosa, and that may be some of the barriers between that you're encountering?
Yes, that's definitely something we're excited to think about. I think in the let's say this Bacteroides vulgaris example is a good one, because sometimes you can catch those two clades in the same person. So, sort of rules out, not ever being the same person, but you could still say maybe they're segregated along the intestinal tract. There we think that if it really was just lack of contact, it would just change the overall rates and not change the sort of lengths of the transferred fragments. And so that extra little bit of info has us thinking, maybe it has more to do with restriction modification systems or potentially things like that. And so that's something we're sort of currently trying to think a lot about.
Thank you, great talk. Have you looked at whether certain types of sequences have higher recombination rates or transfer rates? So for example, potentially amino acid repeat regions or any other sort of sequence-based similarity rather than species grouping?
Yeah, let me just sort of go back to this plot because this might help. So, one thing we have kind of looked at is where on the genome they are getting transferred. And so, to first approximation, I would say they get transferred everywhere across the genome. But, if you look at it closer, you actually do see some systematic differences. And the sort of thing we're trying to figure out now, I guess, is how much of this is driven by just differences in the recombination machinery, right? Like sequence characteristics or regions of homology or lack thereof, versus how much is actually driven by selection, right? So one way to get sort of rates of realized recombination that have less to do with the abundance of the species and more to do with something that's constant is if most of these things are actually positively selected on their own. And so we're trying to sort of pair this data where you can like see the accumulation of individual stuff with the sort of opposite thing where we like look at the differences between all of us and look for regions that are shared. And so you can find pretty cool examples of regions that are shared amongst lots of people that kind of overlap with some of these peaks here, and that suggests that maybe they're being driven by ongoing selection for certain functions that are encoded.
Yeah, or also the disappearance of regions.
Or the disappearance of regions.
Extinction.
Yeah, the dirty secret here is that in order to do this sort of thing in metagenomes, we have to ignore all the interesting parts of the genome, the variable regions, and so we hope to sort of go back to this eventually with isolates, because that's where we think most of the selection would be coming from, are the regions that disappear or pop back. Cool. Thanks. Thank you.
Okay. We made it. Thanks everyone for hanging in there all day. I think I'm not going to try to summarize all that data, but just a few highlights. So, I think one thing that I'm really left realizing is that, the methods for genomics and metagenomics have really matured dramatically over the last 15 to 20 years. And so these methods are really robust and rigorous, and that's amazing to see. I think hopefully the other thing that you can take away from today is that the microbiome field is a lot bigger than genomics. And so, we heard about other types of omics approaches, but also, a lot of experimental tools that people are using, and those have really advanced very far. It's also been exciting to see how a lot of the questions that we normally think about in terms of biomedical science are also being tackled in the environmental sphere, and also in all sorts of other systems like beetles, which I think we all want to work on now. And also, the importance of thinking beyond bacteria. We heard a lot about, how cool protists are, and archaea and viruses. And so, it's really exciting to see that the field has really branched out in terms of the microbial diversity that's being studied. And then, finally, and maybe most importantly, I think you should all take from this that California is where you should be. We've only really scratched the surface of the microbiome research in California. We haven't even really sampled all the labs in the Bay Area. There's many more that hopefully will come to future events. And, there's a whole Southern California that I hear exists. So, it really makes me feel fortunate to be here and to be in this community where people are so excited about the science and also very collaborative. And so with that, I really want to thank our speakers again. Maybe you can join me in. Thank you to ETS for keeping the lights on, Dorothy Wang. Our amazing administrative support, Nancy Cook and Kristen Dolan. And with that, please join us out in the mezzanine for a reception, and we hope to see you all again soon.